Good afternoon, everyone. Welcome to our virtual international conference on food digestion. My name is André Brodkorb and I'm based in the Teagasc Food Research Centre in Moore Park.
Giblin go Codal International Food Digestion is Linda Giblin, also from Teagasc. Over to you, André.
Thank you, Linda. There's Linda's Irish introduction, one of the two official languages here in Ireland. As you can see, we are not live from, or I'm not live from the from Moore Park, but from my living room, because we are still in the COVID situation, and really all of you should have been here, right here in Cork, right now, enjoying the beautiful sunshine that we have every beginning of May. That's why the conference is at the beginning of May, and enjoy the Irish hospitality. But here we are on another online virtual event, but we have a great program. So before I forget it, I would like to thank the organizing committee, especially Linda Giblin, Myrne Egan, who is doing the magic in the background with all the webinars and the YouTube and all of this, all the session chairs that have selected the talks, and last but not least, Nathalie Lemar in Rennes. Without you, this event couldn't have happened. So I have a number of slides. We might as well dive in. And here we go. Okay, so we have 7 sessions, 3 sessions this afternoon, followed by a poster session, and our very capable Moore Park postdoctoral team are running it. They have volunteered and they are running it quite independently. And tomorrow morning, we start early in the morning, 7:30 Irish time. So all times are Irish time, Dublin time, with a live session from Australia and New Zealand, followed on by 3 more sessions. And at the end of it, we have a couple of prizes. Please register to all 3 events. So I just put them down here, all 3 events. So obviously you are registered already on day 1, but you have to register for the poster presentation as well. It's a different login, and tomorrow morning is a different login. Now, we also live stream on YouTube. So if you go to our YouTube channel, the Infogest channel, we stream live, and we are very excited about it. It's the first time we are doing this. Or if you scan this QR code, you go directly there. So we had 57 abstract submissions from 4 continents, 17 countries, really good. And today and tomorrow we have 28 oral presentations from 13 countries. And I'm particularly delighted that we have 22 PhD students. We also have 24 flash presentations, 3-minute flash presentations organized by our Moore Park postdoctoral team. The book of abstract is available either on the LinkedIn page. So there's an event page, and if you go there and look for the Food Digestion Conference, you can find it downloaded there, or you go to the Cost Infogest website and look in activities, and you find it. Tomorrow, at the end of the conference, we award prizes. The best oral PhD talk of all talks. So we have 2 judges working in the background, watching all these talks. It's a lot of talks. It's 12 hours talks, pretty much non-stop. And we also have the best flash presentations. The prize for the best oral PhD talk is sponsored by the Tim Company. It's a cash prize. And the best flash presentation is a free registration to next year's conference.
So thank you, André. So as André mentioned, we had to postpone our 7th International Conference on Food Digestion because of the pandemic. So we're holding it next year in Cork, in Ireland, on the 3rd to 5th of May, 2022. So Cork is in the south of Ireland. It's an old city founded in the 14th century on islands in an estuary. So the conference will be held in the Maryborough House Hotel, and the gala dinner will be in the renowned Ballymaloo restaurant, which is the best of Irish food. So please come and join us.
Thank you, Linda. So I hand over to Didier Dupont, who is the chair of the Infogest Network. And Didier, I have your slides, so just tell me when I have to go to the next slide.
Okay, thank you very much, André and Linda. It's a real pleasure for me to welcome you at the virtual International Conference on Food Digestion. I wish to thank, of course, our friends from Tagas, from Ireland, for making this event possible. And before we start with the scientific program that looks extremely exciting, I would like to say a few words on the Infogest network that is organizing all these kind of events. So the Infogest network, next slide, is the international network on food digestion, as you may know. Why is it so important for us to understand digestion? Well, you all know that there has been a dramatic increase of the diet-related pathologies over the last 30 or 40 years. So there is really an emergency to prevent the development of these pathologies rather than having to cure them. When we ingest food, the gut plays the first interface between the food and our body, and digestion will provide nutrient to the body, and we also release molecules that can have a positive or negative effect on human health. However, the mechanism of food disintegration in the gastro-intestinal tract remains unclear. So we strongly believe that if we are able to better understand the mechanism of food digestion, we will be more aware of the effect food can have on our health. So the scientific objective of the network are to compare the existing digestion models, try to harmonize the methodologies, propose guidelines for performing new experiments. We also wish to validate in vitro models towards in vivo data, develop in silico models, also develop models that are specifically adapted for specific populations, such as the infant and the elderly. We also try to identify the compounds that are released during food digestion, and we try to determine the effect of the food matrix structure on the bioavailability of nutrients and bioactive molecules. But of course, in order to do that, we need to gather people from very different horizons, different disciplines, such as food science, nutrition, gastroenterology, and so on, in order to share and improve the knowledge that we have on food digestion. So this is how the network looks like at the moment. We are a little bit less than 600 scientists coming from 45 countries. So all the continents are now present in Infogest. And you will see on the next slide that there are a lot of companies, private companies, that are following the events that we are organizing and the activities that we have in the network. Almost 60 companies are involved in Infogest. And you can see also on the next slide, this is how we are organized. We are organized into 6 different working groups working in parallel. And you will hear a lot about the people who are leading this group during the next 2 days. So the network, of course, is open to everyone. It's completely free. You can come in, join the network, and opt out whenever you want. So if you are interested to join us, just send me an email. You can see here my email address. So next slide, just a few information regarding some major outputs that we had over the last years. You are probably all aware that we released a consensus static in vitro digestion model through a paper that has been extremely well cited in 2014. Then we updated the model more recently in a paper that has been published in Nature Protocols, and it's a hot paper with already 293 citations. And finally, I would like also to mention that we have released a book in open access, so you can just go on the Springer website and you will have access to this book that will give you an overview of the in vitro and ex vivo models in order to understand and study digestion. Well, as Linda told you, we are all organizing every 2 years the International Conference on Food Digestion. So this is an event that is followed by 250 or 300 people all over the world. And as Linda told you, next slide, the next conference will be organized next year between the 3rd and 5th of May 2022 in Cork, Ireland. So I wish I will see you there. And I hope that you will enjoy the conference. So have a great conference and see you in Cork next year. Thank you.
Excellent. Thank you, Didier. Thanks a lot. I think we are a good team. So we have 12 hours of oral presentation, which is a lot. So we have sessions and we have 20 minutes breaks in between. We decided to do it like that simply because we join from different time zones, so time is really irrelevant. We record the entire conference recorded and we put it on YouTube afterwards. So if you miss a talk, you can look at it later. So because there's so much 14 hours of presentation, you know, there's a bit of fun stuff we introduced. We have a hashtag. So this is a hashtag virtual international conference for food digestion 2021. If you are active on Twitter, please tweet. And on top of that, we have a couple of interesting things that Murren introduced. One is a Mentimeter. So if you go to menti.com and put in this code, and this code will appear later on other slides, we ask you a couple of questions. One of them is, from where are you joining today? Because we actually don't know from where you're joining us today, because all we have is your email, and sometimes there's gmail.com or Hotmail or whatever. We don't actually know from where you are joining us. So if you go to menti.com, put in this code or scan this icon, we will end up with something like that, and we see where you're from. And later on or tomorrow, we have another poll where I asked you what kind of topics you would like in the future Infogest webinars, because since the lockdown, since September, we had a number of webinars and they're all published on our YouTube channel, and they have been quite successful, and we still want to keep it up, and we have to keep it up for another while. So we're looking for topics that are of interest to you. Okay, so we can actually move on now to session 1, and the first session is on food digestion and in vitro digestion, and it is based around the Infogest Working Group 1, and the co-chair is Lotte, Egger, and Isidra, who are also the working group leaders. If you can show yourself already, Lotte and Isidra. Just 2 points of ongoing activities. Lotte initiated an international ring trial on the static Infogest method because there's a drive to bring this to a next level, to an IDF and ISO method. And there are 30 laboratories involved, and that's really coordinated by Lotte. And a second international ring trial is ongoing, where the next step, basically the semi-dynamic Infogest method, is being evaluated, and we have 11 laboratories involved, and again, it's coordinated by Lotte. Okay, so without any further delay, we have 6 talks. We have 1 senior speaker and 5 PhD students. I would like to introduce the first speaker, Amélie Declaire. Can you show yourself? There you are. So Amélie is the first speaker. All talks have been pre-recorded, just out of safety. And afterwards, we have a live question and answer sessions. At the bottom of your screen, there's an icon Q&A. You can type in the questions, and we will ask the presenter, in this case Amelie, the question right afterwards. So we have a few minutes for question and answers. So I hand over to Amelie.
Good afternoon, everybody. So I will present you today a work that has been conducted in STLO in Rennes and in collaboration with PNCA, a lab in Paris. And I will talk today about the endogenous nitrogen contribution to in vitro digestibility of animal and plant protein. So regarding the nutritional quality of dietary protein, two parameters are important. First, the essential amino acid composition in regards to the requirements that can be easily determined thanks to total amino acid analysis, and also the protein digestibility that needs to be determined either in vivo or using in vitro model. This is a proxy for nitrogen or amino acid bioavailability. By combining these two parameters, we can determine the protein digestibility corrected amino acid score. So protein digestibility in vivo can be determined after digesta collection, particularly at the terminal ileum, which is more accurate. By calculating the difference between the total nitrogen collected in the digesta with the nitrogen ingested, we can thus determine the nitrogen that has been released by digestion and then further absorbed through the intestinal epithelium. However, within this nitrogen collected at the end of the terminal ileum, there are different origins. Of course, there are some dietary nitrogen remaining that has not been digested, but there are also some nitrogen originating from endogenous origin. Previous papers have shown that there are a lot of bacterial origin in this nitrogen endogenous, but there are also some nitrogen coming from enzymes, from epithelial cells, or from the mucins. So different type of digestibility can be determined. If no correction is applied to the total nitrogen as determined in the ileal digesta, this is called an apparent digestibility. If a correction is applied but based on basal endogenous nitrogen losses, for instance determined with a protein-free diet, this is called a standardized nitrogen digestibility. And if the nitrogen endogenous losses are accurately determined, that is with the dietary nitrogen also accurately determined, thanks to 15N labeled dietary protein, we can call it a true digestibility. So the more correction we do, the more accurate we get, and we can increase the value from apparent to true digestibility up to 20%. As we all know, we need to reduce the in vivo trial for ethic but also cost constraints. So as we all work with in vitro model, our goal is to evaluate a protein digestibility using in vitro models. So with in vitro models, we know exactly how much endogenous nitrogen we insert in our model. However, we don't know which fraction will become digestible during the digestion. In other words, we don't know how much autolysis of this enzyme will occur during digestion. So the digestible nitrogen in vitro can be determined either for some authors using the soluble nitrogen, or other authors have also used the molecular weight cutoff of 10 kilodalton, which will be used here in the present study. So the present question here is to evaluate in vitro whether there is a contribution of this endogenous nitrogen to the digestibility values. So we used different 15N labeled dietary proteins to follow precisely the fate of our dietary nitrogen and to evaluate accurately the endogenous nitrogen losses. So we used micellar caseins or gluten at 3 different dietary protein levels, 4, 8 or 16%. We also used a real food matrix, a RUSC, with a 4% dietary protein content. And we also did a blank digestion, that is, that only enzymes were inserted in our solution. We submitted this protein to in vitro static digestion using the Infogest protocol, and we did a gastric and intestinal phase with different sampling time. We used porcine pancreatine plus bovine bile and porcine pepsin as enzyme. Then we collected our digesta and we did some fractionation. First, we did a centrifugation to remove any insoluble material, and from the supernatant, we performed an ultrafiltration with a molecular weight cutoff of 10 kilodalton. After complete recovery, we freeze-dried our digester and then measured the total nitrogen and 15N enrichment to evaluate the true digestibility. We also performed size exclusion chromatography on the soluble nitrogen in order to evaluate whether our ultrafiltration was efficient or not. So we did different calculations. First, by collecting all the digestible nitrogen in regards to the nitrogen in the meal, we could determine an apparent digestibility. By considering solely the dietary nitrogen, we could determine a true digestibility, which was thus corrected for endogenous losses. We also tried to determine a standardized digestibility, which would be the digestibility corrected only for the nitrogen of a blank digestion, so even before digestion, and fractionate below 10 kilodalton. So now let's have a look to the results. So first, we can have a look at the contribution of the different sources of nitrogen. So first, we can see that there is a great contribution of the nitrogen coming from the pancreatine, so endogenous nitrogen, especially when we have a low dietary protein content. So it's almost half dietary nitrogen and half endogenous nitrogen. Of course, by simple dilution, this proportion of endogenous nitrogen will be reduced when we increase the dietary protein content. So the question was whether after digestion we would still find as much endogenous nitrogen as before digestion. And we were quite surprised by seeing that actually still 40% of endogenous nitrogen was contributing to the total nitrogen found in our digestible part, which is the fraction below 10 kilodalton. And again, of course, because we increase the dietary protein content, this contribution gets lower. As I said, we performed a size exclusion chromatography in order to evaluate whether we efficiently remove the fraction above 10 kilodalton. And so here is the profile before ultrafiltration, and here we can see first that actually the fraction in the soluble nitrogen is quite small, above 10 kilodalton. After ultrafiltration, 90% of this fraction is removed, so there is an efficient removal of this fraction. And even between 10 and 5 kilodalton, 50% of the fraction will be also discarded. So the real molecular weight cutoff is lower, but this is as expected. And here we also checked when we had a higher level of 10 kilodalton fraction that there was an efficient removal in the gastric phase, and there was also 90% of this nitrogen above 10 kilodalton, which was removed by ultrafiltration. Now let's have a look to this endogenous nitrogen fraction below 10 kilodalton compared to what was inserted at the beginning in the digestion flask. So here we observed that it amounted up to 40, almost 50% of the nitrogen which was inserted at the beginning of the digestion through the enzymes, mainly pancreatin. And what we could see is that there was a difference between the dietary protein content, especially a greater amount of this endogenous nitrogen below 10 kilodalton, which was greater at 4% of dietary protein within our solution. So now let's have a look in comparison with a blank digestion. Here we could see that the blank digestion, so even before starting the digestion, there was already almost 40% of endogenous nitrogen below 10 kilodalton. And this proportion increased, especially for this 4% dietary protein content. However, this increase of about 30% was lower than in the presence when the digestion was conducted as a blank, which means with no dietary protein. With no dietary protein, the blank led to an increase of this fraction of 50%. And this was in agreement with the fraction observed in the free amino acids. So here we can say that there was a more pronounced autolysis of the enzyme when the low dietary protein content was digested. If we take a look at this bar here, it is the RUSC. So the RUSC is a real food matrix, and actually it has an intermediate behavior with no increase of this autolysis. So now that we had a look at this endogenous contribution compared to the total endogenous nitrogen, let's have a look on the impact on the apparent digestibility. So here there was a great difference between the dietary protein content, and also values were largely above 100%, especially for low dietary protein content. When correcting for the endogenous losses, we reduced greatly these values, especially for the 4% level. There was still a small difference between the dietary protein level for the gluten only. And when digesting the RUSC, a real food matrix, the impact was also reduced. So here there is a great contribution of the endogenous nitrogen losses on the digestibility value. Now let's have a look to the standardized digestibility. So standardized digestibility is what we would calculate if we did not have any 15N labeled protein. I mean that we would have done a correction with a blank at T0 and collecting the nitrogen below 10 kilodalton. So here you have a reminder of the true digestibility, and when we correct simply by the blank with no only the nitrogen below 10 kilodalton, you have the result in red. We can see that for the 8 and 16%, the correction is very good because the true digestibility is in agreement with what we have found. However, for the 4% dietary protein content, the correction was not sufficient and led to an increased value of digestibility. But this was actually artificial, as this was only due to too low correction for endogenous nitrogen losses. So, as a conclusion, we have seen that the correction for endogenous nitrogen is very important to get accurate digestibility value. it is not negligible on the proportion of digestible nitrogen, particularly when a solution of dietary protein with low content is digested, and this was the case mainly for the 4% dietary protein content. However, we have seen that when dietary protein were included in a food matrix, this impact was much reduced. So we can discuss also about this 10 kilodalton cutoff with this membrane filtration that we all know will induce some loss, but this could be improved by using the profile determined through size exclusion chromatography. Or even, as we have seen that the fraction above 10 kilodalton was relatively low, we could also work on the soluble fraction released during digestion. We need to go further with the amino acid digestibility, as this is important to evaluate the DIAS. DIAS is the digestible indispensable amino acid score, which is the new recommended index by the FAO. So we need to determine amino acid digestibility either in the soluble fraction or in the 10 kilodalton fraction recovered by different means. So I want to thank you all the collaborators involved in this work, particularly Lucille, who conducted this work during her internship. And I also want to thank you for your attention.
Fantastic, Amelie. Great talk. Thanks a lot. So you're live on air now after listening to your own talk, which is usually painful. I don't know about you, but usually is. So we have a number of questions. The first question is from Anne Reeder. Why was a cutoff of 10 kilodalton for digested proteins chosen?
So I know this molecular weight cutoff can appear a bit high, but this has been quoted by different papers, for instance, by Mugan in quite a while ago. But so why did we choose this molecular weight cutoff? It's because we know that we are missing the enzyme from the epithelium, so the brush border enzyme. And also, based on our profile, our SEC profile, you can see that the digestion does not seem to be very complete. So that's why we chose this cutoff. In addition, we have internal data that shows that the digestibility values determined with this cutoff were well in agreement with in vivo digestibility. So I could also complete by maybe the soluble nitrogen, which is also used sometimes in different papers, could be also another approach.
Very good, very good. We have a few more. The next one is from Lionel Bovetto. Could we conclude that autolysis is not constant between pure enzymes and food matrix type? So does the food matrix affect the autolysis of the enzyme?
It seems that there is a different behavior between dietary protein alone with water and the food matrix. And this, I think, can be completed by a different lab with the next talk. But so, yes, I agree. I just have a remark between pure enzymes. I don't know if it's a typo or not, but we use pancreatin. So it's, as you all know, it's an extract of the pancreatic juice of pig. So it's not pure pancreatic enzymes. And this may actually affect also the results because I'm pretty sure there are many different origin of the nitrogen within this pancreatin.
Yeah, I have one more question by Claire Gaudichon, so I better read it out. If you can answer that very briefly. So in endogenous losses in vivo, only 50% is composed of amino acids, including protein-bound amino acids. have you any idea of the nature of the non-amino acid nitrogen?
We have not deeply investigated that in our losses, but there might be some urea, ammoniac, but not, I don't know, in our pancreatine. It's kind of, I'm not sure we have that in pancreatines. It would be surprising. So I guess this is more for the in vivo. Yes, it's for in vivo.
Very good.
Other nitrogenous compounds that are not from proteic origin. But yeah, in the paper from Miner Williams, there might be some more information on that.
Very good. So I have a number of questions, but you can actually type these questions now and answer them, Amelie, and then everybody will see the answer. Before I finish off, before I hand over to Isidra, I have a question to you. Did I spot you in one of the images with a nasal gastric tube? Was that you, Amelie?
Yes.
There you are.
When I was in the lab of Claire Godichon.
Fair play to you. Fair play to you. Okay, very good. Thank you, Amelie, again. Yeah, Isidra, I hand over to you.
Thank you. Thank you, André. Very good, Amelie, for your talk. I'm happy to introduce our next speaker. This is Raquel Sousa from Agroscope, and she will continue with the same subject about protein digestibility and DIAS values. So please, Raquel, the floor is yours.
Good afternoon. My name is Raquel Sousa and I am a PhD student from Agroscope, Switzerland. And today I will present some results from my thesis where protein digestibility and DIAS values were calculated using in vitro method. This work is supervised by Lotte Egger, Reto Portman and Isidra Recio from Madrid. Some years ago, FAO has published the report you can see on the left side requesting to have digestibility values provided to all foods. In detail, they requested to determine true allele digestibility using humans. At the same time, they wanted as well to validate the use of animal models to replace human trials. As a last point, they would like to develop and validate in vitro methods for determination of DIAS values, which is the digestible indispensable amino acid score. The in vitro assays allows us to test protein sources that would not be accepted by humans or pigs, and at the same time, it's also possible to mimic infant, elderly, or pathological conditions. Apart of this, the in vitro assays are much more cheaper and without ethical problems. Thus, we develop an in vitro digestibility method, which is based on the static in vitro digestion model from Infogest. This is a protocol which tries to mimic the human digestion inside the falcon tube. All the physiological conditions were taken into account for the development of this method. This method was further validated for its reproducibility by an interlaboratory trial and for its biological relevance by a pig trial. Our method was developed on the frame of Proteus, which is a project founded by Global Dairy Platform and led by Paul Morgan and Susan Hodgkin from Reedit Institute, New Zealand. In this project, they are performing pig and human trials for DIAS calculations. They are using 7 different protein sources. 4 complex foods, namely peanut, black beans, pigeon, peas, and all-bran cereals, and 3 isolated proteins, namely whey protein, zein, and collagen. In our project, we are digesting the very same samples in vitro in order to compare and validate our results toward the in vivo. In this slide, we have represented the behavior of the food during digestion. On the left side, we have represented the food before digestion, where there are mainly intact proteins, and on the right side, we have the food after digestion. After the intestinal digestive endpoint, we mainly see free amino acids and small peptides, but there are also bigger peptides and intact proteins present. We have it color-coded, so the greener a molecule is, the more absorbable it is. It's generally accepted that 3 amino acids up to 3 peptides are actively transported through intestinal walls. Larger food-derived peptides were also detected in blood, but this is mainly due to diffusion. As our model lacks the brush border membrane enzymes, we consider polypeptides smaller than 10 amino acids as being bioavailable, which in vivo would be further degraded by the brush border proteases. For separating the absorbable from the non-absorbable components, we tested different methods and we end up with the best method being the precipitation with 80% methanol. After this precipitation, the pellet represents the undigested fraction and the supernatant the digested or absorbable fraction. To characterize the fractions, we have set up a size exclusion chromatography method where the big molecules elute first while the small molecules elute later. This method was tested with different peptide standards of various sizes, and as you can see here, polypeptides of 8 to 10 amino acids elute around 40 minutes. This limit corresponds about to 1000 Dalton. Like that, every molecule eluting later than 40 minutes is smaller than 1000 Daltons, and therefore we consider them as absorbable. Here I show you the precipitation fractions of 4 example foods from Proteus, 2 complex foods and 2 isolated proteins. In these chromatograms, the gray line corresponds to the pellet, which represents the undigested fraction, while the black line shows the supernatant. As you can see, the peaks of the supernatant are eluting after the 40 minutes cutoff, while the peaks of the green line mainly elute before 40 minutes. Therefore, we can conclude that 80% methanol precipitation indeed separates efficiently the absorbable from the non-absorbable components. On this slide, we show you how we measured and calculate digestibility. After precipitation of the digested food, we end up with a pellet and supernatant. Because there are a lot of digestive enzymes added to the digestion, we need a good enzyme blend to subtract. To control the enzymatic autolysis, we digested the protein-free cookie in parallel to each food. This cookie, only containing starch and fat, proved to be a better enzyme blend than the water digestion. On the right side, you see the 3 different analytical endpoints performed for all these 4 fractions. We determined total nitrogen by Kjeldahl, we measured primary amines using OPA, and the individual amino acids were quantified using HPLC. Before the latter measurements, and in order to release and detect all the amino acids, we performed a hydrolysis with 6 molar HCl. By measuring the total nitrogen, primary amines, or the sum of the amino acids, we can calculate the total digestibility. With the values from the individual amino acids obtained by HPLC, we can calculate the individual digestibilities. The digestibility is then calculated by subtracting the supernatant of the cookie from the supernatant of the food, divided by the total of the food minus the total of the cookie. Additionally, we have defined the cookie pellet as minimal. So, if the pellet of the food minus the pellet of the cookie turns negative, we set this to 0. This avoids the rare cases of having digestibility values higher than 100%. The diet is calculated as displayed here. First, we have to measure the content of each individual amino acid per gram of food protein. Then, we take, for example, the amount of the indispensable amino acid lysine, and we multiply it by the lysine digestibility calculated using the formula from the last slide. This results in the amount of digested indispensable amino acids per gram of food protein, the so-called DIA. The FAO provides a table with recommended amino acid scoring patterns for infants, children, adolescents, and adults. The measured DIA value is compared with this table. So, by dividing the measured DIA by the reference DIA and multiply it by 100, we get the DIAS values. Normally, only the lowest DIAS is reported as the limiting amino acid. Now, you probably wonder how our method performs. So, in this graph, we show you the total digestibility of the protease substrate and we compare it with the in vivo results. Here, I have to mention that these in vivo values are preliminary and they might change in the future. In this graph, the blue bars represent the in vitro digestibility based in total nitrogen. The pink bars, the OPA values. The green ones, the digestibility based in total amino acids, and the yellow bars are the in vivo results. As you can see, in general, the three methods give similar results, but the in vitro value seems to be slightly higher than the in vivo. We also did the comparison for the digestibility of the individual amino acids. In the graph, the blue bars show the in vitro values of the different foods, while the orange bars show the in vivo values measured in pigs, the gray bars are the human values, and the green bars come from rats. As you can see, we obtained similar results and patterns for the in vivo and in vitro assays. The values present here come from triplicate digests, and the error bars are quite small except for zein. This is a very hydrophobic protein coming from corn, which forms clogs inside the digestion tube, causing inhomogeneous results. We statistically compare the in vivo and in vitro methods with the Blunt-Altman test. On the x-axis is represented the average of the digestibility of each amino acid determined by the different methods. The y-axis represents the difference between these averages. The line in the middle of the graph shows the bias between the methods, with the lines of the ±2 standard deviations above and below. The calculated bias for the difference between the two methods is 0.7%, which is really good. And as you can see, all the statistical points except one are located within the ±2 standard deviations as desired. Like that, we can conclude that the in vivo and in vitro digestibility methods are in agreement. In this slide here, we have the final calculation of the DIAS values. Again, we compare the in vitro values with the in vivo ones. For all the foods, we have in vivo peak data to compare, and for whey, protein, and zein, we have also human data available. And we can clearly see all three giving similar results. These results show the great potential of the in vitro digestion as a screening tool for DIAS assessment. Once again, I have to remind you that this in vivo data is preliminary and it might change at the end of the project. For detailed comparison between the in vivo and in vitro DIAS method, we show you the correlation between them in the graph on the left side. We obtained a correlation slope of 0.93, which is very close to the idyllic slope of 1. On the right side, we have again a Blunt-Altman graph comparing both conditions with a bias of 5.3. With that, we can state that we have a very good correlation between in vitro and in vivo DIAS values. So, to conclude, I have shown you that these in vitro conditions provide a good estimate of the in vivo situation for total digestibility, digestibility of individual amino acids, and DIAS. The ADAPT Infogest in vitro digestion protocols seem to be suitable for measuring digestibility of food. We have to state that so far this protocol was only tested with 7 different protein sources, and therefore, in the future, it has to be validated with more foods. As soon as we have tested a significant number of foods, we will see if a correlation factor is needed to perfectly fit the in vitro to the in vivo data. We can also consider including a standard food with a well-known digestibility to each set of digestion as a normalization. As a last point, the recommended amino acid scoring pattern from FAO is giving values for infants, but then is measuring the digestibility in adults. Our model in vitro could be adapted to infant physiological conditions, allowing like this to have a better estimation of these DIAS values. So with that, I want to thank my supervisors for all the support and thank you very much for your attention. And I would be happy to answer all your questions.
Thank you, Raquel, for your excellent talk. Very, very good. So we have again a couple of questions here. I will try to read some of them. Alan Javier Hernandez is asking why using a fat and starch control, and if this sample is comparable then to your foods, the foods that you are using for digestion.
We are actually using only cookie, like protein-free, because we are interested in evaluate the protein digestibility. So if you were adding like more protein, then we would not be sure from where it was coming from. So that's why we took only carbs and fat. And if it's comparable or not, if it's talking about the amounts, I added 1 gram of cookie to each digestion because it was according with the protocol. But for the other samples, I was normalizing it by protein content. So I was using 4% protein content per each digestion.
Yeah, there is another question about how do you prepare the foods for the in vitro assay, if you grind or dried the samples before.
Well, 2 of them, they were cooked, the black beans and the pigeon peas, they were boiled, and the rest, they were used like raw. I just tried to mimic a little bit the chewing, so I was breaking them into smaller pieces, but apart of that, no extra treatments.
Yeah, you have more questions about the cookie, the composition of the cookie of this blank, so you can answer them later on. I think there is another question comparing what do you recommend to use methanol then, or the 10 kilodalton filtration that it was proposed in the previous talk, if you want to follow the Infogest digestion. Do you know the composition of your methanol supernatant?
Yeah, and I mean, in the slide where I was showing the SEC chromatograms, we could see that after the precipitation with methanol, most of the supernatant was coming or eluting after 40 minutes, most of the peaks. So that means they were smaller than 1 kilodalton. So that should be the cutoff for the methanol. So if you are using a filtration with 10 kilodaltons, it will not be the same, of course, because you will have bigger particles in. But I think it really depends for the purpose, what you want to analyze. For us, because we want to determine the digestibility, it's really important to have only the small particles on the supernatant. So the methanol, I think, is better.
Yes, you have another question here about methanol. Why using methanol rather than trichloroacetic acid? But
we tried actually, but the methanol was the solvent that was giving us like better chromatograms. There were no additional peaks, and the cutoff was really like straightforward and very clean. So we took that because of this. The TCA was giving a huge peak in the middle of the chromatogram.
You have many other questions now to answer, but I think that it's better to be on time that we move to the next speaker. Thank you, Raquel. Excellent. Thank you.
Thank you.
Lottie, are you there? You might have some technical issues, so we move on to the next speaker, Ruan Deng from Wageningen University. Excellent, you're live there. Okay, we hand over to you, and we see you later at the question and answer session.
Hello, everyone. Today, I'm very glad to tell you about how we could make use of magnetic resonance technique to monitor gastric digestion. I'm Ruo Xuan, a PhD student from Wageningen University. As most of you already aware of, food digestion is a very important process for nutrients to be available in human body. Among the oral gastric intestinal tract, actually gastric digestion is one of the very important part because it involves both physical breakdown with the muscular construction in the stomach and also chemical breakdown with the help of gastric acid and enzyme like gastric lipids and pepsin. Only when the food particle is smaller enough can go into the next digestion stage. Gastric digestion is important especially for solid food because it can help to reduce the particle size and help to break down the food structure so that the food particle can be more accessible in the following digestion and absorption stage. Moreover, it is the start of the protein digestion. Gastric digestion is very important. With the help of everybody working in this field, actually a lot of in vitro models have been developed. With the available analyzing technique, we could withdraw samples from the in vitro digestion system and then measure the food property and digestion fluid change. Also, we could study the interaction between the food particle and the digestion fluid to better understand the analyzing mechanism. So it does have a lot of advantages. For instance, it's a very well-controlled and simplified system, and then we can nicely design some simple experiment to examine our hypothesis. However, when you think about in vivo condition, actually there are more biological variation, and it is more complex and more flexible. So with the in vitro model, it's quite hard to represent the feedback mechanism, for instance, like how the neural system and how the hormone response taking place. So that arises the question how we could make use of the in vitro digestion result to represent the in vivo condition. Well, with the in vivo digestion, actually there are also some techniques available to directly study. Like you could withdraw samples from the stomach, and you could do some blood sampling to measure the nutrients and hormones, or you could ask the participant to rate their appetite. But all these approaches are either too invasive or indirect. So we were wondering if there is a way that could help us to link the in vitro and in vivo digestion. Well, when you read the title of my presentation, you might already get the answer that we propose. Yeah, we propose that we could use MRI to link the in vitro and in vivo digestion data and it can help to validate the in vitro model with the help of MRI. So for those who are not familiar with MRI, it is an abbreviation of magnetic resonance imaging. This is how it looked like. Well, actually, this was me with one participant. Later, we scanned the stomach of this participant. So why would MRI be this candidate? First, I would like to show you some MRI scans. Well, this is a baseline scan where the participant went through overnight fasting. So you see the stomach was so small here because it was only some gastric fluid in the stomach. And after he drank a glass of milk, you see how the stomach expanded. So with this type of scan, you could analyze the volume of the stomach content, and then you could get an idea on how the gastric emptying is happening. Moreover, when you look at the stomach, you could be able to see what is happening in the stomach. So it also provides opportunity to study the intragastric processes. In addition to use the MRI as a camera to study gastric digestion, there are also some potential parameters that we could further investigate to see if they can also be used to study digestion. We have published a review paper on this last year, so you could find more information over there. But today I would like to draw your attention on the relaxation parameter T1 and T2. So what is T1 and T2 relaxation? Well, because of time limit, I won't go too much into the physics, but I want to let you know that T2 and T1 relaxation are based on the proto-relaxation. And in our study, we observed H proto-relaxation. And the difference between the T1 and T2 relaxation is that T1 relaxation is called longitudinal relaxation time. It happens in that direction. While transverse relaxation time, the T2 relaxation, happens in XY plane. And there are also terms of relaxation rate, which are actually inverse of the time. So we also have the R1 and R2. And in this presentation, I will use the term of the R1 and R2 in the following part. So to make it easier for you to understand why the T1 and T2 relaxation can be useful for digestion, then I draw a transverse relaxation decay of the gastric juice and protein gel. Now you do see the huge difference between each other, and by calculating the relaxation rates value, you could also see directly here that they are quite different from each other, which shows that then we could maybe make use of this to look at different components during digestion. And you might have the question on what other factor would affect the R2. Well, actually, to make it simple to understand, R1 and R2 are dependent on the environment of the proto. If the proto is in a more free environment, then you would have a smaller R2 and R1. If it's in a more rigid environment like protein gel, then you would have a longer or bigger R2 and R1. So actually, there are multiple factors that would affect R2 and R1, like the pH, the concentration of macromolecules, the food structure, and also the water distribution, water content. Well, all these factors are also important during digestion, or are also changing during digestion, which makes the R1 and R2 interesting to be investigated to see if we could make use of this parameter to indicate the change during digestion. Since there are multiple factors would affect relaxation rate, we decided to use a well-controlled and more simplified system. So we use the static gastric digestion model, and we use whey protein gels as our model food. We measure digestion with MRI and also NMR, but today I will only show you the readout with MRI. So this is how it looks like, MRI R2 map. Well, actually, this is a cross-section of the sample, so it is a slice of the tube. On the top, it is the gastric fluid phase. In the bottom, it is the whey protein gel phase. So now you could nicely see how different are R2 values for these two phases, which nicely confirm with what we found from the relaxation decay from the last slide. So how does it change during digestion? After 90-minute digestion, you do see that in a liquid phase, the relaxation rate become bigger. Well, since this is well-controlled system, we know that pH remain no change. So the only thing would happen is that the protein gel will be digested, and then the peptide would be released from the gel phase into the liquid phase. So we also measured the concentration in the liquid phase to know how the protein is digested. By combining this protein concentration in the liquid phase and also the R2 value of the liquid phase, we nicely see that indeed we can use the R2 of the liquid phase to predict the protein concentration in the liquid phase during digestion, which enable us to use the R2 of the liquid phase to predict the degree of protein digestion. We have published this readout in this paper, so you can find more information over there. But I guess now you might have a question in your mind that in vivo condition is not like this. There are more variables. The pH is changing, there are influx of gastric fluid and gastric emptying. So while this approach still be feasible and there are more dynamic setup, this was also the question we had at that moment. Therefore, we decided to step further. So we developed this MRI-compatible semi-dynamic gastric simulator, where we could include this gastric segregation and gastric emptying by these pumps. And I want to highlight that we use airflow to simulate gastric mixing instead of using magnetic stirrers, because we want to put the whole stomach chamber into the MRI scanner, and you cannot put any magnetic material in the scanner. Well, but the airflow works perfect well, so we are very happy with this setup, because then we can do the digestion in the stomach chamber, in the scanner, with the gastric segregation and emptying happening at the same time. So we use this setup, and then we measure the digestion of 15% and 20% whey protein gels. We measure the digestion with both NMR and also MRI to measure the relaxation rates, R1 and R2. And we also measure the pH and protein concentration in the liquid phase to know how the digestion is happening. Now I would like to show you some results. So with the digestion of 15 and 20% gel, here you see how the protein concentration in the liquid phase changes with the digestion time. So for both of them, the protein concentration increased over time, and with 15% whey protein gels, the increase is faster because the digestion is faster, which confirms also with our static digestion model. We also measure the pH, which shows the change during the digestion. We also measured R1 and R2 during digestion. This is a readout from NMR with a digestion of 15% whey protein gel. So for both R1 and R2 during digestion, they were increased over time. The increase could be contributed by the protein concentration increase in the liquid phase, but also pH might be another factor that affects R1 and R2 value. So if there is a correlation between R1 and pH and protein concentration, well, actually we found a nice correlation between the R1 and protein concentration and pH in the liquid phase. And same applies for R2. So when you look at this equation group, actually it shows that R1 and R2 are functioned by the protein concentration and pH during digestion. So with this equation group, if we have R1 and R2 as measured nuance, then we could be able to calculate the protein concentration and pH during digestion. So we decided to examine this with the readout of 20% whey protein gel digestion. So with the equation groups that we got from the last slide, and then we now have the digestion readout of 20% whey protein gel. So we would have the input of the R1 and R2 during digestion of 20% whey protein gel. And with this equation group, we can then calculate the protein concentration and pH during digestion for the 20% whey protein gel. So how does the result look like? Now you could see that actually the estimated protein concentration and measured protein concentration of 20% whey protein gel during digestion are quite close to each other. Same applies for the pH. So this is really a very exciting readout because it shows that although the condition is more dynamic, we could still make use of R1 and R2 to predict protein concentration and pH during digestion. We would also like to confirm the readout with MRI. Here I would like to show you some MRI images. At time 0, there was only gastric fluid in the compartment. Well, from time 5 minutes onward, you see two phases. So the gastric fluid phase and also the protein gel phase. And you would also see that during the digestion, the R2 value of the liquid phase increased during digestion. We analyze also the average R2 value of the liquid phase, and here you could see the trend, how it increased during the digestion. But we also need to measure the R1 during the digestion with MRI so that we could see if we can get some similar equation group where we could use R1 and R2 to calculate the protein concentration and pH change during digestion. With all the promising results from the in vitro experiment, we would like then to step further to see if we could also use the MRI parameter relaxation rate to monitor in vivo gastric digestion. We have designed a randomized crossover trial to study how the different protein gels digested in human body. So now the trial is ongoing. I hope soon I will start to analyze the data and can tell you more about how we could use R1 and R2 to monitor in vivo gastric digestion. This brings us to the end of this presentation. During the presentation today, I have shown you how we could use magnetic resonance parameters relaxation rate to monitor the pH and protein concentration during digestion. And MRI is a very promising method to monitor in vitro semi-dynamic gastric digestion. And for future work, in vivo performance of this method still need to be further explored, and we are now working on it. I am now in the last year of my PhD. I hope soon I will have a nice PhD booklet where I will tell you more about how we could use MRI, this promising method, to study digestion and how it can help us to link the gap between in vitro and in vivo digestion. I would like to thank all my supervisors and other people who helped with the research. And I would like to thank the organizer giving us the opportunity to share our research to all of you. And thank you all for your attention.
Isidra, are you there? I think we have a few issues.
Yeah. Yes, I'm here. Yeah, wonderful. Thank you for your very interesting talk, Rusan. Again, we have several questions here, for instance, about the procedure, how long it takes to measure the T1 and T2 in vivo.
Oh, thank you. Thank you for the question. Oh, actually, this is a very important thing that we need to consider when we conduct the in vivo trial, because when we do the measurement, we need to let the participant hold their breath during the scan. So you cannot make the scanning time for 10 minutes or something, then people would die. So for our sequence, we have like, for the T1 measurement, we have like several seconds for one inversion time. And then we repeat the different inversion time for several scans. So for each scan, it only takes for at longest 4 seconds for the T1 scan. Well, for the T2, it takes around 18 seconds. So they are both fine for the participant to hold their breath.
There is another question about the mixing, because you use air bubbling to simulate the peristaltic movement. So it is suggested that maybe it works for weak or easily disrupted gels, so very weak gels. But how do you think that is going to work with harder structures?
Yeah, thank you. I think this is also a very good point. Actually, the reason why we call our system a semi-dynamic model is because that we call it dynamic because we involve the gastric secretion and emptying, and then we call it semi-dynamic is because we think with our bubbling mixing is not sufficient to really mimic the movement of the stomach wall. But I think that's something that we could improve in the future by using different material or different way of grinding. Indeed, I think if we have the meat, something like meat, then it's quite hard to just using the bubble thing to grind the meat. But for now, with this is the first step of creating a system that could be compatible with MRI. So we start from this simple system. But indeed, in the future, there are still a lot of things to be improved and to be worked on.
Okay, there is another question. If you know if there are already in silico models in the work that you are doing, or are you aware about this? If there is already some in silico models to simulate what you are doing or not?
Well, actually, I think with the in silico model, it's nice to have some in vivo data to support. So with our work, we now are collecting, especially with the in vivo trial, we are now doing a lot of stomach scan. So we hope that our data will be future useful for doing more, not only analyzing the experimental data, but also contribute to better knowing how for instance, how the gastric secretion is working, how the gastric emptying rate is when the food matrix are different. So I think indeed with our data set, it will be it can contribute to the in silico model modeling as well.
Okay, thank you. Thank you very much for you can answer then the rest of the questions online, just to be on time. So thank you for your presentation. Thank you very much.
Thank you.
André or Lottie?
Lottie,
André, shall I? Okay,
you go ahead, Isidra, go ahead. I think
okay, then we move to the next speaker, that is Melanie Tassadak from INRAE in France, and she will present about simulated digestion of biopolymer matrices for assessing active molecules release. So Melanie, thank you, and the floor is yours.
So hello everyone, my name is Mélanie Tadassac and I'm a PhD student at the French National Institute for Agriculture Research in Nantes in France. And I'm going to present you the simulated digestion methods we used on biopolymer matrices in order to assess the release of active molecules contained in it. So this work is a part of my PhD project, which is presented here. So the goal is to study the biopolymer materials with specific porosity, designed by numerical modeling and then 3D printed in order to control the release of active molecules. So this comes from the fact that different models with the same density but with different porous structures can have different release profile kinetics. So the question we are trying to answer is what is the link between the porous structure of delivery systems and their molecules release kinetics. So to do that, we start from the biopolymer that we mix with an active pharmaceutical ingredient, which is an active molecules that we make into objects that have a certain release profile. And this experimental data will allow us to create a model that is able to predict the release profile from the structure of 3D printed objects, and also able to design the structure of 3D printed objects from a targeted release profile. So the first step of this study is to obtain experimental results. And this presentation is going to focus on the method we used to study the release from biopolymer matrices. So this is the outline of the presentation that I will follow, starting with an introduction of the biopolymer we used, which is zein, and its thermoplasticization. So the zein is a biopolymer extracted from maize kernels. And so after fractionation, milling and separation from starch, it represents 3 to 5% of the initial mass of the kernels. It's an amorphous biopolymer containing both polar and hydrophobic domains. It is composed mostly of alpha zein, which has a molecular weight of 20 to 25 kilodalton. And thanks to its amphiphilic properties, it can be plasticized by various compounds, like for example glycerol. And here you can see an example of zein plasticized by glycerol in order to make thermoplastic filaments at 130 degrees. But here, instead of glycerol, we use an ionic liquid as a plasticizer. This ionic liquid is a salt of two active molecules that are lidocainium and ibuprofenate, which are respectively a local anesthetic and an analgesic. And it has been showed by Chaunier et al. That the powder blend of zein and this ionic liquid can be extruded at 130 degrees to make filaments. So the matrices we are going to use as a model in this work are thin films obtained from the powder mix of zein and the API ionic liquid, which is melted under pressure at 130 degrees during 2 minutes. These are the same conditions used as to obtain the extruded filaments. And here you have a photo
photos of the films we can obtain. So now I'm going to present this simulated digestion method we used on our films. So this method is based on the Infogest method, which is a standardized in vitro digestion method generally applied for food digestion. So we put the films into a digestive media and incubate them at 37 degrees. The digestive media firstly represents a gastric phase for 2 hours and is composed of 1 ml of SGF, simulated gastric fluid, with pepsin, which is a gastric enzyme. Then the digestive media is changed for an intestinal media for an additional 2 hours, composed of 1 ml of SIF, so simulated intestinal fluid, with pancreatin, which is a mix of intestinal enzymes. And we also did digestions without these enzymes as a control. So in total, the simulated digestion lasts 4 hours, so 240 minutes, with the following time points. So 5 in the gastric phase and 2 in the intestinal phase. And we use 1 sample for each time point. So from this experiment, I'm going to show you how we studied the molecules' release kinetics. So for each time point of the digestion, we take out the supernatant and we use an HPLC dosage method to dose the lidocaine and the ibuprofen released in the media. The advantages of this method is that it separates the 2 species and allows to measure their concentrations at the same time. So here you can see an example of a chromatogram. We have from a supernatant, and you can see the 2 distinct peaks representing the lidocaine and the ibuprofen. So from that, with the calibration curve and with the area under the peaks, we can calculate the release rates of the 2 molecules during the digestion. So the release rates are in percentage, but without a unit. So on the left-handed side, you can see the release of lidocaine, and on the right-handed side, you can see the release of ibuprofen. You can see the gastric phase at the beginning, followed by the intestinal phase. The orange curves represent the digestion with enzymes, and the blue curves represent the digestion without enzyme as a control. And what we can see is that the release of the lidocaine is superior with digestive enzymes, but not for ibuprofen. And what we also can see is that the ibuprofen is not released during the gastric phase, but only when we switch to the intestinal phase. And this shows a low solubility of ibuprofen at pH 3. We also studied the macroscopic shape and the water uptakes of these matrices. So first, you can see here an example of the macroscopic aspect of the film after digestions. And in order to evaluate the swelling, we measured the surfaces of the films during digestion by image analysis. And we hypothesized that the surface is representative of the swelling. So the image analysis consists in photos we take at each time point of the digestion. And from these images, they are transformed into binary images. And then a program is able to count the white pixels in order to extract information such as the surface, the diameter, and the perimeter. And this data can then be converted into meters with the resolution of the photos. So this graph shows the evolution of the surface during digestion, And we can see that it expands up to 2.5 times the dry matrix. But experiments need to be replicated in order to confirm these results. So we also measured the water uptakes of the film by evaluating the difference of mass before and after water evaporation. And we can see that during the digestion, it increases up to around 70%, which correlates the surface expansion and the swelling we saw before. And these results also need to be replicated. So now, in order to build a numerical model, we need information on the mechanical properties. And this is what we are doing now. We are performing compression mechanical tests on the films and recording the stress of the films versus the strain. And it seems that there's a logical evolution of the mechanical properties during the digestion. So finally, we wanted to check whether the matrix was degraded during digestion and if it was solubilized in the digestive media. So we performed migration on electrophoresis gel with the supernatant to see if there was zein in the supernatant. So here you can see an image of the gel. In orange, it's the digestion with enzymes, and in blue, it's the digestion without enzymes. And what we can see is for the digestion with enzymes, there are only bands with corresponding to the enzymes. So here pepsin, and here the pancreatin, and you have the control here. And for the digestion without enzymes, there are no bands in the samples. So what we can say is that there are no bands of zein detected in the supernatant, or at least it's below the detection threshold. And this is why we concluded that the zein matrices were not eroded by the digestion. So in conclusion, what we did in this study is optimize a protocol to mimic the digestion in order to study the release kinetics of molecules in the gastro-intestinal tract. We also optimized dosage methods in HPLC, and we concluded that the release was by diffusion mechanisms more than matrix degradation, since there was very few zein in the supernatant after digestion. And now the prospects are to perform total protein dosage in the supernatant, so BCA, in order to calculate the amount of protein found in the supernatant. It will be to study the solubility of the ionic liquid in simulated digestive fluids, so especially for ibuprofen in the gastric fluid. It will be to continue the study of the mechanical properties during the digestion, and in order to complete the information on the molecule release kinetics, we need to perform curve fitting, modeling, and determine the diffusion coefficient. And later, it will be to apply all these methods to extruded filaments and then future 3D printed delivery systems. So I just want to thank Anne-Laure Regel for her help to obtain the results on the image analysis, and I thank you for your attention. And I'll be pleased to answer if you have any questions.
Thank you, Melanie, for this excellent talk. Now it seems to work, my mic. I think you are hearing me. So there is one question in the audience that he's asking, or she, if you have looked at how much of your loaded API is released throughout your gastric and small intestinal phases, and especially to complement your release rate data. You have switched off your microphone, I think.
Sorry, yes. So I was saying the release rates are calculated during the gastric phase and the intestinal phase. The release rate I showed in my presentation.
Okay, thank you. And what is the influence of swelling on release of compounds?
That's what we are trying to study, but we think that the swelling increases the release of the compounds from the matrices. And this is what is mainly causing the release of the compound.
Okay. I think, thank you for your talk. And so we switch to the next speaker. André is, I think.
Very good. Thank you, Melanie. Thanks again. Very nice talk. So we move on to the next speaker, and Mariana Radomirovic from the University of Belgrade in Serbia. Hi, Mariana. Good to see you. So I hand over to you, and I see you later during the live question and answer session.
Good afternoon. My name is Mirjana Radomirovic. I'm a PhD student at University of Belgrade, Faculty of Chemistry in Serbia. And the title of my presentation is Ficosinobilin modified beta-lactoglobulin is more resistant to pepsin and pancreatic digestion. This is only a small portion of the results that I obtained within my PhD work that could actually fit into the topic of this virtual conference. For a short introduction to the topic, I'll say a few words about BLG and PCB. Bovine beta-lactoglobulin is the major whey protein with many technofunctional properties desirable for the food industry. As some of you may already know, it is a soluble over a wide pH range, has ability to aggregate and provide structure to foods, and also has great emulsification properties together with its water binding and foaming activities. Additionally, its proteolysis generates peptides with numerous biological activities. BLG has been known to bind many ligands in both non-covalent and covalent manner. And due to its high resistance to pepsin digestion, in the recent years, BLG has often been investigated for the possibility to be used as a stable carrier for incorporation and efficient delivery of bioactive food components. Speaking of bioactive food components, ficosinobilin is a tetrapyral chromophore of ficocyanin, which is a chromoprotein derived from the blue-green alga Arthrospira platensis. There are numerous studies showing health protective effects of ficocyanin, such as antioxidant, anti-inflammatory, neuroprotective, and hepatoprotective effects, all of which have mainly been ascribed to ficocyanin's chromophore PCB. In the work previously published in our group, we showed that PCB covalently binds to the free cysteine residue of BLG in a wide pH range, but more importantly and more relevantly, it is also able to bind to BLG under simulated gastro-intestinal conditions. That said, in the picture on the left side, you can see SDS page of BLG and BLG PCB incubated for 2 hours and at 37 degrees at pH 7.2 and then run on the polyacrylamide gel and afterwards stained with zinc. So intensive fluorescence at around 18 kilodaltons corresponding to the mass of BLG monomer clearly shows that the protein has PCB attached to it. On the right side, you can see SDS page of BLG and BLG PCB incubated in simulated gastrointestinal conditions. Experimental conditions were the same as those previously described in the harmonized protocol by Minekos, except that the addition of enzymes was omitted. So the idea was to investigate whether BLG PCB adduct is able to form in simulated gastrointestinal conditions. We mixed equimolar ratio of BLG and PCB with simulated salivary fluid, and after 2 minutes, the obtained mixture was mixed with simulated gastric fluid for 2 hours, followed by mixing with simulated intestinal fluid again for 2 hours. And at every stage of digestion, aliquots were taken for SDS pH analysis, and after running electrophoresis, the gel was stained with zinc. So as you can see, the BLG-PCB adduct can be formed at all stages of protein passage through gastrointestinal tract. We further evaluated changes in protein secondary and tertiary structure upon binding of PCB by using CD spectroscopy. As you can see, the shape and amplitude of CD spectra in the near UV region indicate the tertiary structure of BLG is slightly changed upon binding of PCB. With that in mind, further evaluate the effects of covalent BLG modification by PCB on some functional properties of protein that could affect its usage in the food industry. We evaluated antioxidant properties and heat-induced changes of BLG and BLG-PCB. So just briefly, BLG-PCB had enhanced antioxidant activity and oxidative stability. It also has a reduced tendency toward heat-induced polymerization, aggregation, but also fibril formation with thermal stability similar to that of the unmodified protein. As for the other functional properties that are of high relevance for this conference, we investigated in vitro pepsin and pancreatic digestibility and the IgE binding properties of PCB-modified BLG. BLG and BLG-PCB digestibility was assessed by simulated gastric and intestinal in vitro digestion. In vitro pepsin digestion of BLG and BLG-PCB was carried out in simulated gastric fluid at 37 degrees, and digestion was followed for 6 hours. Aliquots were taken from digestion mixture at different time points. And the 0-point time sample was also prepared for BLG and also for BLG-PCB. We also had pepsin autoproteolysis controls at 0 and 6 hour time points, as well as protein stability control, which was just a protein without pepsin. This gel was scanned, and the band intensities were densitometrically analyzed using software, and plotted again against the digestion time. So first, as you can see in this figure, a covalent BLG-PCB adduct is more resistant to pepsinolysis than its unmodified counterpart, and as seen by this longer persistence of 18 kilodalton bands throughout the time course of digestion. As I already mentioned, intensities obtained after software analysis of this gel were plotted against digestion time in order to estimate protein's half-life. And the half-life of BLG was estimated to be 14.2 minutes, while half-life of BLG-PCB is 41.3 minutes. Similarly, in vitro pancreatic digestion of BLG and BLG-PCB was carried out in simulated intestinal fluid at 37 degrees, and digestion was followed for 4 hours. Again, aliquots of digestion mixture were withdrawn at different time points, as shown here, and analyzed by SDS pH. And again, of course, 0 time point controls samples were prepared together with pancreatic autodigestion and protein stability control. After SDS page, the gel was scanned and the band intensities estimated using ImageJ software with BLG or BLG-PCB band intensity in 0 time point taken as 100%. So as you can see in this figure, the covalent modification of BLG by PCB again attenuated digestion of BLG, but the effect for pancreatic digestion was not as pronounced as the effect on gastric digestion. Densitometrically obtained intensities were presented relative to the intensity of BLG or BLG-PCB band in 0 time point, which was marked as 100%, and plotted against digestion time. We estimated half-life of BLG to be 3.6 minutes, while half-life of BLG PCB was 5.7 minutes. So, as BLG is one of the main allergens of milk whey, after examining pepsin and pancreatin digestibility, we were interested in examining IgE binding properties of BLG and BLG PCB, as it has already been known that covalent modifications of BLG, for example, upon glycation and modification with polyphenols, can influence its allergenic potential. So the assessment of the IgE binding properties of this important food allergen upon PCB modification is of great interest when considering its usage in food industry, especially having in mind all those enhanced properties of the protein that I mentioned previously. So for the purpose of investigating IgE binding properties, we use Western blot and inhibition ELISA to study binding to milk allergic patients' IgE. So as seen in this figure, Western blot probed with a serum pool from human subjects allergic to milk show that both BLG and BLG PCB bind IgE from the milk allergic serum pool. Additionally, the IgE binding properties were assessed on a quantitative level by inhibition ELISA. Unmodified BLG was first coupled to the plate, and then IgE binding of milk allergic patients' serum pool to the BLG was inhibited by either BLG or BLG PCB, and the bound IgE was detected using anti-IgE antibodies. The concentration needed to inhibit 50% of the signal obtained in the absence of inhibitor was calculated, and it was used to compare the IgE binding potencies of the samples. As seen in this figure, both proteins show similar, virtually the same pattern of dose-dependent inhibition of milk allergic patients' IgE binding to the unmodified BLG, with their inhibition plots almost overlapping. That indicates that BLG modification by PCB results in no significant changes in IgE binding potency. And indeed, we calculated IC50 values of BLG and BLG PCB, and they're shown here. And there were no significant differences in these values. So to conclude, covalent modification by PCB significantly affected both pepsin and pancreatin digestibility of BLG, with the PCB modified BLG being slowly digested in both cases, but particularly with pepsin. The unaltered IgE binding capacity of BLG PCB in comparison to that of unmodified BLG is an important feature of BLG PCB, especially when considering its possible application in the food industry. The high resistance of covalently modified BLG to pepsin and pancreatin could thus be used to efficiently deliver bioactive PCB to some distal parts of the gastro-intestinal tract, and therefore contributing to gastro-intestinal tract health. Also, this lower tendency of the modified BLG to enzymatic digestion could enable not only prolonged delivery of PCB, but also of bioactive peptides originating from BLG itself. And finally, I would like to acknowledge the help in funding from the Ministry of Education, Science and Technological Development of the Republic of Serbia, Belgian Special Research Fund, Serbian Academy of Sciences and Arts, and also the project Food and Twin under the European Union Horizons 2020 Research and Innovation Program. I would like to thank you for your attention, and I'm open for your questions.
Very good. Thank you, Mariana. Very good talk. We have two questions. One is from Pete Wild from the Quadrum Institute in UK. And he asked, normally, a native beta-lactoglobulin is fairly resistant to pepsin hydrolysis. Had your beta-lactoglobulin been subjected to any heat treatment?
Yes, it has been subjected to a treatment of 2 hours during the preparation of covalent adduct, but nothing more than that.
And what kind of heat treatment was that?
It was 2 hours at 37 degrees.
37?
Yes, 2 hours at 37 degrees.
Okay, yeah. And
So
was it
no,
Sigma beta-lactoglobulin? Did you buy it? Or?
No. We isolated it itself. We purified it itself.
Okay, so no heat treatment whatsoever, really.
No. Just 37 degrees for 2 hours.
Okay. The second question, Augusto Santos. I would like to know if the research was performed with other times in the gastric phase or only 6 hours, and if this set of time could interfere on pepsin digestibility results with the complex.
Okay, so we had different time points during digestion, and we took the aliquots of digestion mixture during the digestion that lasted for 6 hours. So we followed. We had time points at 2 minutes, 5 minutes, 15. I would have to see my presentation again to see exact times, but we followed it during we followed it at different time points.
Yeah. One last question from myself. So the PCB or PC is a pigment, right?
Yes.
So it absorbs and possibly some kind of emission. Did you use that absorbance and emission in any way to follow the interaction, first of all, and the digestion?
So for the digestion, we did not include emission of the PCB, but during the studies of kinetics of binding of PCB to BLG, we used emission of PCB. We used spectropolarimetry to study kinetics of binding.
Excellent. So thank you again, Mariana, and I hand over to Lottie. Welcome back.
Thank you. So for
never a dull moment, online.
Now I would like to introduce you to Ji-Hong Cheng from Chagas, Ireland. So he's working in André Brodkorb's lab, and he will talk about membrane filtration reduces the heat load of infant formula and affects the semi-dynamic infant in vitro gastric digestion.
Hello, everyone. My name is Yi-Hong Chen. I'm fourth-year PhD student from Chagas and Master Technological University. Today, I'm going to introduce you a part of my project, use membrane filtration to increase the native whey protein content in infant formula, and show you how it affects infant formula digestion in the gastric models. For someone not familiar with infant formula processing, let me introduce standard wet processing with heat treatment. This is a brief scheme of making infant formula powder by using wet processing. Processing of infant formula always introducing several of heat treatment for purpose of microbiology safety or removal of water. Heat treatment results protein denaturation may cause some physiological and healthy concern. For example, infant formula compared to breast milk has significant different gastro-intestinal transits and digestion kinetics. Specifically, infant formula has doubled gastric empty time compared to breast milk. Denatured protein may lead to allergy because change of protein morphology and forming advanced glycation end products. Moreover, consuming infant formula made by high heat treatment may result high blood urea. These concerns are all directly caused by dietary protein. Therefore, reduce the natural level of protein is important. Membrane filtration technology are well used in dairy industry to reduce the heat loading during the processing. Our project is about introducing membrane filtration into the infant formula processing. So in the following slides, I will call our product as MEM infant formula. This is the scheme of MEM IMF. Skim milk mixed with whey protein isolates then feeds into a 1.4 microfiltration unit. This recirculated flow processing was able to retain over 90% bacteria, but without effect of protein in the permeate. Permeate will further feed into a 0.2 microfiltration unit. Retentate from 0.2 microfiltration contains casein micelles and residual bacteria was mixed with other ingredients, underwent a heat treatment for sterilization. Permeate from this membrane were considered as filter sterilized. Two sterilized solutions merged in a mixed tank, then sent to dry processings. The two treatments have different thermal loading. Therefore, let's see their protein profile in the infant formula powder. The inner layer indicates water-soluble and insoluble materials. Membrane-filtrated infant formula contains significantly higher soluble materials compared to heat-treated infant formula. The molecular weight of whey proteins and aggregations in infant formula are shown in mid-layer detected by cytochrome HPLC. The membrane-filtrated infant formula contains less large protein aggregations, but more smaller particles indicated to monomeric proteins. A notice at here, monomeric protein contains their native and denatured monomeric form. Thereof, the denatured form can join the acidic aggregation at the pH level of stomach. I will talk about this later. It is more apparent to show in a graph of HPLC absorbance. MEM infant formula has apparent peaks indicate to monomeric, BSA, beta-lac, and alpha-lac, while these peaks are absent in the HT infant formula. The outlier shows the amount of native whey protein preserved in the powder. Membrane-filtrated infant formula preserved 18.3 of total whey protein remain in their native form, while heat-treated only has 2.5%. They're significant difference, but both of them look slow, right? It is because the trial proceeds in the lab scale. By the scale up, it can significantly increase the native whey protein in the powder. The detail I will talk about later. Now, two types of infant formula have been made. To investigate will preserved native whey protein affect digestion, we set same dynamic infant in vitro gastric model. Infant formula was based on international consensus same dynamic in vitro digestion model, with adult parameters changed to infant level. Gastric half-emptying time was set at 78 minutes for infant formula according to the previous research. The ratio of food and secretion was 30:20. Enzyme activity and secretion were lower than adult parameters. There's 5 gastric emptying points for stimulating the consecutive gastric empty. After ingestion, both ingestions has pH level slightly below than 7. Videos shows digestion performance in the gastric model. Microstructure of digester was shown using confocal microscopy. Red cycle are fat droplets and the green lines shows proteins. At the second gastric empty point, about 62 minutes, both of infant formulas had a pH about 5.2. In the gastric model, infant formula started forming coagulations. The observation of microscopy shows protein form aggregations around the fat droplets. At the third gastric emptying point, the pH of heat treatment infant formula was lower than the MEM. Member-filtrated infant formula formed small coagulations. Most of them float up to the top of gastric model. In contrast, heat-treated infant formula formed large coagulations in the gastric phase. You can apparently see them in the videos. Microstructure level of MEM-IMF shows protein aggregates are getting smaller compared to the last GE point. At the 124 minutes gastric emptying point 4, both of samples has pH about 3.5. Coagulations of member-filtrated sample tending to flocus, but they are remaining large in the heat-treated samples. Microstructure showed us proteins were almost hydrolyzed in MEM infant formula, but heat-treated infant formula still has significant portion of protein aggregations. At the last gastric emptying point, pH for both samples reached about 2.5 because gastric fluid dilutes the digestion. The gastric model contains member-filtrated infant formula turned to cream, while heat-treated sample still can observe coagulations. The microstructure level fat droplets in member-filtrated infant formula has broken down. Look at the heat treatment sample. Proteins still surround the fat droplet. By observation of macro and micro level, infant formula treated with member filtration shows faster disaggregation of coagulants. So what about their protein degradation and peptide release? Member-filtrated infant formulas R-square of pH linear close to 1 indicate to a smoother pH declaration. Along with declining pH, member-filtrated IMF have significant higher free amino acid group released at all gastric emptying points except the fourth one. Digestats were filtered by 0.0 micrometer syringe filters, then measure their molecular size by Cyclocution HPLC. Columns on left side indicate to intact whey proteins or their casing fractions at the same molecular weight. Column on the right are peptides. Digesta of member-filtrated infant formula has more intact proteins, is according to what we found in the powders. Proteins were continually hydrolyzed along with the digestion. Peptides release reached the peak at GE.4. In contrast, protein digestion of heat-treated infant formula was slow after GE3, and the peptides release reached the peak until GE5. The gel electrophoresis shows casing in member-filtrated infant formula were absent after gastric emptying tray. The strongest band relate to peptides apparent at GE4. So what's the relationship between coagulation formation in the gastric mode and protein digestion? First of all, in the soup net of infant formula feed, it contains free proteins and protein aggregations. Because of the homogenization, the branch protein aggregations embedded into the fat droplet, and also it contains native whey protein. Along with digestion and declining pH, the acidic aggregation occurred. These aggregations are protein-fat complex. The natural whey protein are able to aggregate at pH 4.6, while native whey protein aren't. Native whey protein also cause short branch protein aggregation embedded on the fat droplet during the homogenization. So the native whey protein interferes the formation of larger protein-fat complex. Apparently, the larger protein-fat complex leads to larger coagulations in the stomach. Moreover, native whey protein does not join the acidic aggregation at this level. Therefore, less protein engaged in this reaction results lower density of protein-fat complex. It is the reason why member-filtrated infant formula coagulations flow up to the top of the gastric model. The smaller coagulations made the smoother gastro-intestinal transmit. These smaller coagulations further influence digestion by increased enzyme accessibility. Moreover, the native whey protein lowers their emulsion stability, leads to embedded protein dissociated from fat droplet. Rely on the positive results we got from lab scale. We scale up to pilot scale and in vitro digestion using the same schemes. Scale up. Batch size increased from 15 kg to 900 kg. VCF increased from 3 to 9.5. For anyone who don't know the term VCF, you can simply think more unheated permeate stream in this case. The ratio of native whey protein to total whey protein will also increased from 18% to 60% in the member-filtrated infant formula. Pilot scale infant formula will feed pigs aged 28 days. Each group had 10 pigs. They are fed twice a day and infant formula account for 35% of their total diet. Pigs were slaughtered at age 56 days. After 3 hours, their last breakfast. Comparing molecular size of in vivo digester, pigs fed by member-filtrated infant formula had higher intact proteins and higher peptides in the stomach when they are slaughtered. These results are consistent with in vitro results. Other in vitro results were also comparable to the in vivo. For example, the higher degree of hydrolysis was found in pigs' gastric digester that ate member-filtrated infant formula. Moreover, member-filtrated infant formula released more free amino acids in pig stomach. This result was not statistically significant, but it's understandable because it's still the gastric phase. For more detail, please see the flash presentation number P12 presented by Kohodo. Conclusion: the in vitro same dynamic infant gastric model by modifying the pyramid to infant is comparable to in vivo piglet digestive model. By introducing member filtration to process of making infant formula can enhance the native whey protein content. More native whey protein results infants form smaller coagulants in the stomach and enhance their enzyme accessibility. Smooth digester transmit, stabilize the pH declaration, and reduce the emulsion stability. Combining all these variants, member-filtrated infant formula had higher proteolysis. Potentially, it can accelerate the gastric empty. The gastric kinetics were changed. It is possible that gastrosophageal reflex may be modified because reducing stimulation to the sphincter.
Thank you, everyone. This is everything I want to share.
Thank you for this interesting presentation. There is one question by the audience, and he's asking how you did estimate the 150 minutes for an infant. Is this based on the in vivo situation, or how did you estimate that?
Thank you for the question. It's quite difficult to find the Fulton Fulton gastric digestion time. The gastric digestion time for Fulton baby. So the data was from a review paper called Specificity of Infant Formula Digestion Conditions. Some clues for developing relevant in vitro model, published in 2014.
Okay, and one question from myself. You see that the proteolysis is faster in the MEM infant formula.
Yes.
What size the peptides, are they smaller? Do you have more free amino acids? Did you check on that as well?
We didn't check the free amino acids from the in vitro gastric digestion, but we do find the more amino acid being released from the in vivo model. So for the detail,
so do you plan?
Yeah, for the detail, actually, we can find for more detail. Actually, we can find in the flash presentation by the co-holder. He's going to present more detail about the in vitro model. In vivo model.
Yeah. Okay. Thank you. Thank you. So with that, we are going further. I think, André, I give back to you.
Yeah. Excellent. If everybody else can just show themselves. So this is the end of the first session, and I think it went pretty well. Always some technical issues, but I think that's normal. I think we are, in a virtual world now, and I think we just have to live with that. So I would like to thank the session chairs and all the speakers. I will put on a slide in the background, and we take a break now, and we will continue 2:35, which is in about 30 minutes. We probably start a little bit earlier because it's good to have some extra time. Okay, thank you all, and see you later. And I just share my screen with you. Please don't forget to go to the menti.com, and later on one of the slides is the code that you have to put in. So to put in the location from where you're joining us from. Okay, see you later. Well done, everyone.
Welcome, everybody. So this is session 2, and we're going to introduce our 2 chairs, which is Pascale and Yuri. So I'm going to hand it over to you to introduce session 2 and all of our speakers.
Thank you very much, Linda. And ciao, Pasquale. It's a great pleasure to be here to share the screen with you, although we cannot meet physically. It's also a great pleasure to start.
Same to me.
It's the same pleasure. And it's a great pleasure to welcome a nice cup of coffee and some good food science in the form of this second session. And our first speaker comes to us from all the way from France, which thanks to the screen, you won't feel the distance, right? So I leave the floor to you, Tessier.
Hello everyone, I'm Romain Tessier, a young PhD in the PNCA lab in France, in Paris. And I'm here to present you our work on the protein quality of sunflower in biscuits, ingested together with apple puree or chocolate in healthy humans. In this project, we focused on sunflower as it is a good candidate for new plant protein sources for some reasons. Its seeds have a high protein content, about 30% in oilseed cake. It is already largely cultivated in Europe, and a previous study in rats showed the high digestibility of a sunflower protein isolate, about 95%. However, there is no study on its protein quality in humans. So, the first aim of this study was to determine the protein quality of a 15N labeled sunflower protein isolate incorporated in biscuit in healthy humans. And in the frame of a parallel methodological development of a less invasive method to determine amino acid digestibility, called dual tracer method, sunflower biscuits were co-ingested with either chocolate or apple puree as vectors of 13C labeled amino acids. The meal was composed of 12 sunflower biscuits ingested in 9 meals every half hour. It represented 156 grams of biscuits, including a total of 25 grams of intrinsically 15L labeled sunflower isolates. Alongside, volunteers received either chocolate for a total of 60 grams or apple puree for a total of 100 grams. Chocolate and apple puree are very different in terms of texture, energy content, macronutrient composition, and also, as you can see on this table, polyphenol contents. And we know that anti-nutritional compounds like polyphenol can interfere with protein digestion and metabolism. At the end, 12 volunteers were included, 7 received chocolate and 5 received apple puree alongside the sunflower biscuits. There was no differences between the two groups for anthropometric data, except that there were more women than men in the group receiving sunflower and chocolate. The clinical protocol was on 2 days. On the first one, volunteers were intubated with a nasoileal tube, and the progression of the tube through the intestine in the stomach was checked with X-rays. On the second day, volunteers received the 9 meals for 4 hours, and we sampled ileal content, plasma, and urine. Ileal content continuously and pulled by half hour, plasma every 30 minutes for 4 hours, then every 1 hour, and urine every 2 hours. The samples were made for 8 hours after the first meal ingestion. All these data allowed us to determine ileal digestibility of nitrogen and of individual amino acid, to determine also the incorporation of dietary nitrogen in urine and plasma, and to calculate 2 protein quality indices, the digestible indispensable amino acid score, DIAAS, and the net postprandial utilization, NPPU. The first results were ileal nitrogen flow kinetics, both endogenous and exogenous. For this figure and all other figures, the group received sunflower and chocolate will be represented in black, and the group who received sunflower biscuits and apple puree will be represented in gold. In this figure, you can see the endogenous nitrogen flow expressed in millimole by 30 minutes. As you can see, there was a difference between two groups and a higher endogenous flow with chocolate compared to apple puree. For the exogenous nitrogen flow, which came from 15N sunflower biscuits, there was no difference between groups. At the end of the 8 hours, there was no difference either in the accumulation of exogenous nitrogen, and consequently, there was no differences between groups for sunflower real ileal digestibility of nitrogen, RID. It was 86.1% when co-ingested with chocolate and 87.6% when co-ingested with apple puree. Altogether, it's a relatively low ileal digestibility compared to other plant and animal protein, determined also in humans. We also determine ileal individual amino acid digestibility, presented in the table on the right. Values were variable from glycine about 70% to methionine about 93%, and there were also a high inter-individual variability present in both groups. Altogether, this data showed that the ileal digestibility of sunflower is moderate. Also, we can see that there are no differences between groups for each individual amino acid, except for the average individual indispensable amino acid digestibility, which was lower when consumed with chocolate than when consumed with apple puree. With this ileal amino acid digestibility, we could determine an index of protein quality, the digestible indispensable amino acid score. It corresponds for each indispensable amino acid to its amount in the protein compared to a reference pattern and corrected by its ileal digestibility. We use the scoring pattern of our older child, adolescent, and adults proposed by the FAO, and the calculated nitrogen to protein conversion factor used was 5.56. As you can see, the DIAA ratio was inferior to 1 only for lysine for both groups. It was 0.97 when consumed with chocolate and 0.99 when consumed with apple puree. That means that the sunflower amino acid composition was slightly limited in lysine, and the ratio was superior to 1 for all other indispensable amino acids measured. Next, we looked at the incorporation of 15N in plasma-free amino acids. As you can see on this figure, representing exogenous nitrogen in millimole by 30 minutes in the plasma-free amino acid pool, you can see that this exogenous nitrogen from 15N sunflower biscuit appeared faster when associated with apple puree compared to chocolate. In the same way, we looked at the incorporation of 15N in urea pool. First, in urinary pool, we can see that there is no group effect as the curves are similar. The incorporation of exogenous nitrogen was progressive and stabilized after 6 hours after the first meal ingestion. And in the body urea, which is plasma urea, we can see on this figure that exogenous nitrogen transfer to body urea was faster and more transient with apple puree than with chocolate. However, at the end of the 8 hours of experiment, there was no statistical differences between the two groups. To finish, we estimated the next postprandial utilization, of which we have the formula indicated here. To simplify, it represents the data in nitrogen retained in the body 8 hours after the first meal. It is the ingested nitrogen minus intestinal and metabolic losses. As you can see, metabolic losses are estimated by urea losses, both in plasma, in body urea, and in urines. As you can see, despite the differences described before between the groups, there was no difference between meals for the parameters, nor for the NPPU score. Moreover, sunflower NPPU seemed good, higher than meat or rapeseed, that was about 70%, but lower than milk, described as 77%. To conclude, sunflower protein isolate has a moderate ileal digestibility, about 86%, compared to other protein isolate, which are usually over 90%, and is slightly limited in lysine. Its NPPU is considered good compared to other results obtained in healthy humans. But however, these results could be due to the repeated meal pattern we used, as digestibility studies are made usually with one meal protocol. For the second aim, we saw that the meal composition, chocolate or apple puree, had an effect on endogenous nitrogen flow, ileal digestibility of indispensable amino acids, and post-absorption amino acid metabolism. But, however, it didn't interfere with nitrogen metabolic utilization. We hypothesized that the presence of anti-nutritional compounds like the high level of polyphenols in chocolate could explain these differences. To finish, I would like to thank all the people involved in this project, both in my lab, PNCA, and our partners. I would also like to thank Sophie Protel, INRAE, and the INRAE for funding this work. And thank you to the organizers for allowing me to present my work today. And thank you all for your attention.
Thank you very much, Romain, for this very well pre-recorded interesting talk. I welcome you on screen. I hope you're well. And we can get started with the question. First of all, you're getting a lot of feedbacks of your nice talk. And people are asking, what were your considerations in selecting the apple puree and the chocolate in terms of polyphenols, because apples also contain polyphenols?
Yes, so the first choice was chocolate, because we added 13C free amino acids in it, so we couldn't have something that was cooked like biscuits. And afterwards, when we saw that the first results with chocolate were not that good, we have to choose fast another vector for the 13C amino acids, and we have to choose a vector that was uncooked, easily available in market store, not too liquid, and we can easily mix free amino acids in it. so we choose apple puree. It's maybe not perfect, but it was the choice we made at this time to proceed with the experiment.
Okay. Yeah. Another question that comes to mind is, did you try to go for isocaloric matching between the products? Because if I noted correctly, there was a big difference in the calories per gram of meal.
Yeah, of course. No, it was not. We didn't try to achieve it, but of course it can influence the results, because chocolate is also very full of calories.
Okay. People are asking, have you thought of using another protein like animal proteins, just to show differences between animal and plant proteins? Have you considered this?
We considered it, but as it's already enough complicated to have one group of people with intubated with nasoileal tube, and we are also in a second study 2 other plant proteins, rapeseed and flaxseed. So we choose to compare plant proteins, but not to compare to effective animal protein.
Okay. Hold on, I'm trying to have a look here at the questions. So another question that comes out is, do you think that the effects of the chocolate or the polyphenols is actually through the interactions with the dietary proteins, or is it by blocking the digestive proteases? What do you think?
We can think that it is an interaction between proteins in the biscuit, so also sunflower proteins, but also gluten proteins that was in the flour of the biscuits. And we can think that it could interact with polyphenol and make them less available for digestive enzymes.
Okay, so there are a few more questions coming up in the Q&A, so please, just for the sake of time, if you can have a look and see if you can type in your answers.
Of course.
And Pasquale, I think the next speaker is for you to present.
I'm delighted to introduce the next speaker. She's Marta Silva from the University of Porto in Portugal. Her presentation is, does substituting Western diet constituents by healthier food impact nitric oxide production upon in vitro digestion? Please, Marta, floor is yours.
Hello, my name is Marta and I'm a PhD student. Today I will be presenting this work named, does substituting Western diet constituents by healthier foods impact nitric oxide production upon in vitro digestion? Non-communicable diseases represent the leading cause of death worldwide, killing 41 million people globally each year, and cardiovascular diseases, followed by cancers, chronic respiratory diseases, and diabetes, are among the leading causes of death and disability globally. Low-quality dietary patterns, such as a typical Western diet, represent a risk to the development of several non-communicable diseases. Diets can also directly influence the inflammatory state, which in turn is also a risk factor for disease. Within the diet, dietary fat and fatty acids have been associated with low-grade inflammation and with both the development and treatment of non-communicable diseases, depending on the fatty acids we're talking about and the food matrix they're in. Dietary guidelines and dietary indexes are very useful, but together with most evidence on diet's effects on inflammation, they are largely based on epidemiological studies. For this reason, it is quite hard to correlate such indicators with laboratory studies, which makes it hard to unveil the mechanisms behind this inflammatory potential of foods and establish causal relationships. Aware of such limitations, the Diet Impact project in which this work is integrated was created, aiming to evaluate the impact of replacing unhealthier foods by healthier alternatives within a whole meal. in this case a typical Western diet meal, and verify the health effects using a foodomics approach, namely through in vitro biomarkers. As an overview of the present work, 6 meal models were obtained by replacing some foods within a Western diet model, and nutritional composition of various dietary indexes were calculated. We focused on fat and fatty acids due to their inflammatory potential. All meals were then subjected to the in vitro gastrointestinal digestion, and after determination of fatty acids content after digestion, bioaccessibility was calculated. Digested meals were treated and exposed to mice macrophages in order to assess the nitric oxide production. These results were correlated with both initial and bioaccessible composition of meals and dietary indexes. Given all this, the main goals of this work consist of assessing the impact of replacing unhealthier foods by healthier alternatives within a typical Western diet meal, through the chemical determination of fat and fatty acids composition of meals, the calculation of the theoretical dietary indexes based on the fat and fatty acids composition, and their ability to decrease the nitric oxide production of mouse macrophages, RAW 264.7, after gastrointestinal digestion. Regarding the preparation of meals, the control meal, a typical Western diet meal, consisted of a fatty beef, French fries, and a sugary soft drink. The meal weighed 800 grams and had around 1,000 calories. The goal was to make real-life replacements in the perspective of the consumer. To achieve the other meals, one replacement at a time was made, aiming to maintain energy and macronutrients as similar as possible. To that, a fatty beef was replaced by either mackerel or chickpeas, or lean beef and olive oil. In other, half of the amount of French fries were replaced by broccoli, and lastly, a freshly made strawberry juice replaced the sugary soft drink. To try and maintain the energy and macronutrient values, food composition databases were used to initial plan, then each meal was prepared, cooked, weighed, and mixed, and the nutritional composition was then assessed in the laboratory through official methods, from which the fatty acids were determined with gas chromatography with flame ionization detection after methyl transesterification. To digest the meals, the static in vitro digestion was performed following the Infogest 2.0 protocol. After gastrointestinal digestion, meals digests were treated before cell exposure. The digests were centrifuged at 12,000 G for 30 minutes, and the bioaccessible fraction was filtered and transferred to a tube with a molecular weight cutoff filter of 30 kilodalton. This tube was then centrifuged for 45 minutes, and the fraction that passed the filter was diluted with complete medium. The dilution used was the dilution 1:3 that was determined based on cell viability results through MTT assay. This treatment procedure aims to further remove non-bioaccessible fraction and remaining enzymes and micellarize fat droplets. The nitric oxide production was assessed in mouse macrophages through the grease reaction assay. Positive controls were stimulated with lipopolysaccharide, and the attenuation of nitric oxide production was calculated as a percentage of nitric oxide production by cells exposed to LPS. Now for the results. Compared to the Western diet meal, despite trying to maintain macronutrients composition and energy values as similar as possible, since these are real food substitutions and the total quantity of meals was prioritized, the nutritional composition varied a little, including the fat content of meals. As expected, fatty acids profiles also varied between meals. Compared to Western diet meal, regarding saturated fatty acids, only the strawberries meal was similar, while regarding polyunsaturated fatty acids, total and omega-3 were all different. Both Western diet meal and mackerel presented similar amounts of omega-6 polyunsaturated fatty acids. With this principal component analysis, based on fat and fatty acids composition of meals, we can see here with principal component 1 that replacing beef with mackerel or chickpeas, which translated in higher levels of polyunsaturated fatty acids, that I will now name PUFA, separates meals the most. Regarding the second principal component, it separates meals regarding the amount of fat or fatty acid and the type of PUFA, either omega-3 or omega-6. Regarding the impact of food replacements on dietary indexes, mackerel replacement presented the lowest thrombogenic index and a high ratio of omega-6, omega-3, and the lowest dietary inflammatory index. Chickpeas replacement led to a lower heterogenic index and ratio of PUFA saturated fatty acids, although a high ratio of omega-6, omega-3. On the other hand, broccoli replacements led to the worst heterogenic and thrombogenic indexes. Looking at this table, seems that replacing half the amounts of French fries with broccoli is worse for health. However, this is not linear at all. Dietary indexes are useful tools, but they should not be interpreted alone, since the impact of whole meals on inflammation and health depends on several other factors that were not considered here. For example, the amount of fiber, bioactives, among others. Bioaccessibility also varied among meals depending on the fatty acids considered. However, the impact of food replacements on bioaccessibility was only significant for saturated fatty acids and in the mackerel meal. Here we can see there was some variation among the results, however the difference were not significant. Here we can see that replacing beef by mackerel or chickpeas significantly attenuated nitric oxide production, being that mackerel replacement was the one decreasing nitric oxide production the most. In this graph, we can see the variation of both heterogenic and thrombogenic indexes and nitric oxide production. We can observe a similar variation trend between these indexes and the nitric oxide production. However, strong correlations with statistical significance were only verified between nitric oxide and thrombogenic index. Similarly in here, despite also observing a similar variation trend between nitric oxide production and fat and some fatty acids composition, a strong and significant correlation was only found for omega-3 PUFA, both initial and bioaccessible fractions. In conclusion, we observed that substituting Western diet constituents by healthier foods impacted nitric oxide production. Nitric oxide production was strongly correlated with omega-3 PUFA and the thrombogenic index. Its production was also significantly decreased when beef was replaced by either mackerel or chickpeas. Both these replacements also led to the lowest heterogenic and thrombogenic indexes of all meals. In addition, the mackerel replacement presented the lowest dietary index and the ratio omega-6, omega-3. Chickpeas replacement presented the highest ratio of PUFA saturated fats. The impact of whole meals on health is complex to study and depends on many factors. Since we verified that replacing unhealthier ingredients with healthier alternatives had an impact on nitric oxide production in mouse macrophages, we now want to verify this effect on other inflammatory markers and cell lines, namely human cells. Therefore, we are performing a comprehensive study of meals' effects on inflammatory markers, also taking in consideration their whole nutritional composition besides fat and fatty acids, through the gene expression of human macrophages after being exposed to meals. With this, we aim to bring some new findings that help uncover some mechanisms to understand a bit more the development of non-communicable diseases. I'd like to finish by thanking you for your time and attention, and I will now take any questions you may have. Thank you.
Thank you, Marta. We are back to the live session. Okay, I can see you. We have time for some questions. I'm reading a question from Fred Barnes. He's asking, okay, it's very similar to what a curiosity I also have. You focus primarily on fatty acid, but there are also large differences between the milks in polyphenols and other bioactives. Can you really uncouple these effects from the effect of nitric oxide production due to fatty acids?
Hi, thank you for the question. Of course, when we are talking about meals, we can't really uncouple the effects. And I think that studying whole meals has this advantage and at the same time a challenge, because here we focused on the fat content and the fatty acids profile. However, we know that this is not the only component affecting the nitric oxide production, of course. So this is a preliminary experiment. So we are now aiming to continue this and also correlate to the rest of the nutritional composition and bioactives as well. Maybe in some time we will have more interesting results to reveal.
Yeah, from myself, I had the same curiosity, but also had there could be, you concentrated on the lipids, of course, but nitric oxide derives from arginine decomposition, from the action of the nitric oxide synthase. So it's did you check if there is any correlation between composition, protein composition, amino acid composition of broccoli, for instance, the presence of arginine or other nitric oxide generating amino acids or so?
Yes, so I do not have the exact values to present because we are still analyzing, but there was always a question that popped up. So we here correlated with fat and fatty acids because we know there is a strong correlation with inflammation, and here the nitric oxide production was a way for us to measure inflammation in this preliminary assay. So we will now continue to measure not only nitric oxide production, but also to see the gene expression, for example, of some cytosins. So we will see. But in the meantime, yes, we will do the correlation with proteins, of course. Also, we have the fiber content that it's not directly we do not directly correlate here because we do the gastro-intestinal digestion, and so we rule out, but we can still correlate to some extent because we know the initial values, and so yes, definitely.
Okay. Then there is a technical question. Did you perform oral phase? If yes, you used which enzymes for the oral phase and the gastric? Very technical question.
Yes,
please.
Yeah, sorry.
Go on.
Yes, so we did all the protocol. So we did the oral phase and the gastric phase. Regarding the exact enzyme, we use the salivary amylase. And for the gastric phase, we use the pepsine and gastric lipase. I don't know if that was the question you were.
Yeah, that was the question. Okay. We have time for our last question. There are many, but okay, I will choose. Okay, there are many comments for your nice presentation. Thank you. But there's a question, would you expect any difference in your results of nitric oxide if you study the effect of bioavailable fraction, for example, using models of intestinal barrier?
So this is a very interesting question because this is actually what we are doing right now. So here, as you saw, we did not perform any transport assays, but this is something we are doing now with the KCO2 HT29, and we will see. I don't know. I think yeah, I think that from what we are seeing so far, the replacement of fatty beef with roasted mackerel and with chickpeas seems to be the one that has the most effects. But maybe we will see something with the broccoli as well.
Okay, there are several other questions, but okay, you please, if you can reply writing to the people that who made the questions. Thank you.
Thank you.
So thank you very much, Marta. Uri.
Sorry, it took me a minute to open the mic. It's a great pleasure to continue onwards with our food digestion session. It's a pleasure to welcome Katarina Smiljanic. I hope I'm pronouncing your name right. If not, then please excuse me. Coming to us from Serbia, from the University of Belgrade. She'll be talking about the use of trypsin as a tool to study protein digestibility. Katarina, the screen is yours.
Good afternoon. I wish all of you happy Orthodox Eastern holidays. My name is Katarina Smiljanic, and I'm going to present today topic trypsin as a proteomic probe to assess food protein digestibility in relation to the post-translational modifications. About the background, is trypsin performance affected by modifications on sessile bonds, and why is that relevant? Well, we know that trypsin is the major intestinal endopeptidase involved in protein digestion, and in the same time it is preferred digestion tool in bottom-up proteomic experiments. It cuts right after lysine or arginine residues, unless followed by proline, and we know that as the Kale rule. Studies based on machine learning of large proteomic data that generated with trypsin revealed the additional complexity of trypsin sequence-specific proteolytic efficiency far beyond known Kale rules. And until present, no one wondered and conducted experiments answering the question if trypsin really does care or does not about different post-translational and chemical modifications. Therefore, we have set up hypothesis and the aim of our study, and that is that porcine trypsin used in proteomics can serve as a probe to decipher differences in sessile bond hydrolysis caused by PTMs based on steric and charge changes introduced as possible hindrance or facilitation to its active site. This is hypothesis number one. If the first hypothesis is proved, we further hypothesize that the effects observed would be even more pronounced with human trypsin, since it is less efficient compared to the porcine counterpart. We have undertaken study on our porcine-derived trypsin-generated proteomic data of the major peanut allergen para-H1 from the raw and roasted peanut to assess possible facilitating or hindrance effects on trypsin digestion efficacy caused by post-translational and chemical modifications, primarily positioned on lysine and arginine residues. We believe that this topic is relevant when considered from the aspect of human gastrointestinal digestion of food allergens and proteins and post-translational modifications introduced by food processing impacting lysine and arginine residues. However, not limited to them solely. Now we come to the methods section, and we can see here what we have done. And the tricky part started when we realized that there are no software globally that can determine or calculate the extent of the miscleaved and cleaved residues within a certain portion of modified and unmodified sequences. Precisely, if we observe here this figure, we can see that, for example, this modification labeled here as D, and this is in dehydroxylation. In this sequence where this arginine is positioned dehydroxylated, we can see that there are modified portion of proteome like 1, 2, and 3, and unmodified, this is the rest of the blue peptides. Now, in the modified portion, there are miscleaved as this one and cleaved. So this has to be calculated, and also the same for unmodified portion. So none of the software can provide you automatically with this data. Therefore, we have developed a manual approach consisted of a frequency count method and relative quantification method via extracted ion chromatography curve areas, which is supposed to be more accurate because it is based on label-free relative quantification, while this is just frequency count. It doesn't take into consideration the abundance. We can now move to the results section to see what. In the results section, the first result presented is alignment of the 3 RAH1 isoforms identified from the gel bands corresponding to the mass of the full-length protein. And we can observe here the all potential sites for trypsin action represented by the numbers above the sequences, where the bold versions of the numbers represent those sites that have been identified in our study. And the blue lines under the sequences, they represent immunoepitopes from RAH1 allergen, while the red squarish represent the modified residues of arginine and lysine identified in our study, and there are 13 of them. In the next slide, we are going to see the zoomed version of this interesting sequence with enclosed with a dashed line. In this slide, we can see the graphical solution that PIC software provides in profiling of post-translational modifications, concretely here in the case of raw and roasted peanut. And the sequence patch is interesting because it represents one of the most potent immunoepitopes of RAH1 allergen, and we can just wonder what could happen if some of these post-translational modifications introduced here can do if it hinders trypsin in doing its action or proteolytic hydrolysis on peptide bonds. This can, of course, prolong the peptides and perhaps increase their sensitizing potential. In continuation, we can see the comparison of the experimentally determined trypsin cleavage efficacy via frequency count approach and the peptide cutter prediction based on certain isoform of RAH1 allergen. Position of lysine and arginine residues corresponds to the data from slide representing the isoform alignment, and gray shades denote mismatch between experimental and predicted data. We wanted to actually stress here the increased frequency of the mismatch data between experimental and predicted one in the modified portion of the RAH1 sequence compared to unmodified residues in RAH1 sequence. Here we have 9 cases of greater difference than 30% in tryptic efficacy, and here only 6 examples among numerous residues, which is of course reflected in the pair testing, t-test done, and we would like to highlight the fact that this is because none of the softwares globally does not sense or appreciate or include the existence of PTMs in its prediction algorithms. So the cause for such greater extent of mismatches between experimental and predicted data is most probably because of modification that happened on these arginine and lysine residues. And which one are those we are going to see in the next slide. Now in this slide we can see the all confidently identified modifications within RAH1 isoform sequences, starting from acetylation, ending by propionamide, and the one that are shaded in gray and reddish color denotes those one who are above the threshold that we initially set arbitrarily for the frequency count approach, 15%, and label free relative quantification approach 10%. Now we can see that there are more cases that support the effect of hindrance in comparison to facilitation effect that are only fewer here. In the next slide, we are going to see the combined approach where we combined all our raw and roasted peanut samples into one category, because here primarily we were interested to see if trypsin sense and react on the existence of post-translational modifications, while to differentiate between raw and roasted peanut is the subject of our next current study. As explained earlier, in this slide we can see statistically significant cases of modification associated residues with a significant difference in trypsin miscleavage, including 4 different positions along the RH1 sequences. Now, out of these 4 are associated with methylation, and also there are formylation and dehydroxylation, and all of them, they support the hindrance effects to the trypsin activity, while only one, and that is methylation of R342 residue, involves facilitating effects. Finally, conclusions, and we can say that data support hypothesis number 1, because out of 13 differently modified lysine or arginine residues, there are 3 residues, namely K248, R259, and R271, where methylation caused significant hindrance to trypsin action by at least double increase in abundance of miscleaved peptides. Similar situation is with dehydroxylation and formylation. Only methylation of R342 suggests facilitation of hydrolytic efficiency, and this observation warrants further dedicated research. Primary sequence context influence is strong. However, influence of post-translational modifications is not negligible, especially in the situations where its saturation is over 5% per residue. It is necessary to develop software that will automatically differentiate and sense and quantitate cleaved and miscleaved sites within the modified and unmodified sequence parts before high-throughput analysis via deep machine learning are possible. Influence of the neighboring PTMs should also be tested and not solely directed to those on lysine and arginine residues. In the meantime, it is possible to start testing hypothesis number 2 by applying methods shown here on the in vitro simulated Infogest 2.0 gastro-intestinal digestion protocol. I want to thank you for your attention and would like to highlight the contribution to this project. We are all from the group of Professor Tanja Cirković-Veličković, and the funding was by Ministry of Education, Science and Technological Development of the Republic of Serbia, including the European Project Horizon 2020, Food and Twin. I thank you once again, and I'm ready for the questions, if any.
Thank you very much. Katrina is with us now live, right? Not recorded, real person, to answer questions.
Real, yes.
Well, first of all, welcome. Thank you for a very, very interesting talk. To start off the discussion, I will continue one of the questions that actually came in the previous session or in the previous talk. And that is, how much do you think that these differences in the methylation these fine subtleties are actually of biological relevance to allergenicity?
Yes, well, this is something that has to be explored in detail. This is very, very initial study, as you could notice, and it did not involve really the data after the, let's say, at least in vitro Infogest or any type of the real digestion protocol that we usually perform. This is one thing. And the other thing is that we have to be aware about the percentage of the certain post-translational modification and chemical modification after food processing. Sometimes this extent is much larger than 5%. And then it's really sensible to undertake such studies, like carbamylation, for example, that we did not bring here into the focus because we are doing in parallel. We have another study, actually it is under review in food chemistry, so I didn't want to share this data at the moment, but believe me, sometimes you have several modifications from the group of from carbamylation and carbonylation, etc., heading to late Maillard products that are really much above, let me tell you, 20-30%, and then it has a sense, all this story, you know. If this was your question.
Yes, actually, because if I remember correctly, about 20 years ago, people were really trying to find the best way to cook or to try to treat allergens, you know, through cooking, boiling, Maillard, you name it, to reduce allergenicity of these foods. But that's the tough cookie. I think it's a tougher cookie than one would expect, because allergens are very potent in terms of their structure. Yes. How much is the analysis sensitive or can highlight structural changes or structural differences, conformational?
Well, this was done on proteins that were completely resolved, I mean, completely denatured, okay? So this is an interesting question. We can, with the bottom-up proteomic approach, I guess we cannot answer to this question. Maybe with the top-down, yes, but also there are so many requirements before you make the protein for mass spectrometry that it's always, you know, very much of tolerance. But you know, we have to start from somewhere. If you agree, we really have to start from somewhere, and not to behave like ostrich digging our heads into the sand and let's pretend that you know, post-translational modification, they don't exist. The point here that I wanted to bring is that you know, we have to acknowledge them, and we have to start to dealing with them and see what they cause. You know, maybe they don't cause so much trouble, but let's be clear with that. if you remember, like one phosphorylation on P53 changes the whole cascade of tumorigenesis. So we have to be, you know, fair with them.
We have to give it our best effort, right? We're in the 21st century, so why not try?
Yes.
I'm absolutely with you. Thank you very much, Katerina. Hold on, let me check again in the Q&A if something new came up. Nothing else came up. Okay, so I take the opportunity again to thank you, Katerina, for the interesting talk, and to invite Pasquale to give us our next speaker for today.
Yeah.
Thank you very much, Katerina.
I'm pleased to introduce the last speaker of this session. She is Silvia Tagliamonte from the University of Naples, and she will speak about profiling the intestinal permeability and well-being status of healthy subjects reporting gastrointestinal discomfort upon milk consumption.
Hi everybody, I'm Silvia Tremonte, a PhD student from the Department of Agricultural Sciences of Naples, and I'm really pleased to introduce you the results of our study about profiling the intestinal permeability and well-being status of healthy subjects reporting gastrointestinal discomforts upon milk consumption. And the reason behind this study is that it has been estimated that about 40% of Western population suffers of functional gastrointestinal diseases associated to food intake. And one of these is called functional dyspepsia and is characterized by early satiety and postprandial fullness or epigastric pain related from meal. And these discomforts do not only affect the gastrointestinal tract, eliciting gastrointestinal symptoms such as nausea, vomiting, abdominal bloating, flatulence, diarrhea, but can also affect the general well-being status and the quality of life, like stress, anxiety, and depression levels, and in turn, the social interaction. And among food-related discomforts, those associated to cow milk consumption without a diagnosis of lactose intolerance or protein allergy are really frequent. And several lines of evidences suggest the role of gut permeability in the onset of food-related gastrointestinal disease. Indeed, one of the major tasks of the intestine is to form a defensive barrier lying between the intestinal milieu and the external environment, in order to prevent the absorption of damaging molecules from the external environment. And this protective function of the intestinal mucosa is called permeability. And there are two key components of the intestinal permeability. The first one that is called paracellular permeability between the enterocytes and is regulated by tight junction, and the transcellular permeability within each enterocyte and is regulated by transporters and channels. However, a condition of intestinal inflammation can lead to a gut leakiness, disrupting the tight junction and increasing the paracellular and transcellular permeability of molecules of different size coming from the food digestion. And because of that rationale, we hypothesized the role of gut permeability in modulating the rate of lactose and bioactive peptides coming from milk digestion, which can cross the intestinal barrier and through the enteroendocrine pathway can affect the gastrointestinal movements and in turn the gastrointestinal symptoms. So we aim to characterize, in this study, we aim to characterize the psychological profile and behavioral attitudes, as well as to monitor the anthropometrical, dietary, and physical activity levels of those subjects reporting milk-related gastrointestinal diseases, and then to determine the implication of the small intestinal and colonic permeability in the occurrence of milk-related gastrointestinal disease. And in order to do that, we screened 101 subjects on the basis of habitual milk consumption and related gastrointestinal diseases. And those potential eligible subjects underwent a lactose breath test in order to exclude from our study those subjects whose gastrointestinal disease could be explained by lactose intolerance. At the end of this selection, we enrolled 39 subjects, of which 20 were habitual milk consumers or HMC group, the control group, which had a milk intake higher than 700 milliliters per week, in comparison with 19 non-habitual milk consumers or NHMC group, which had a milk intake lower than 150 milliliters per week. All subjects were characterized for anthropometrical data, weight and age, and then for emotional states of depression, anxiety, and stress level, eating behavior, physical activity level, and then for the individual perception of quality of life. All volunteers finally underwent a triple sugar probe-based gut permeability test, during which they received a solution containing lactulose, mannitol, and sucralose, and then we monitored their expression in urine within 2 intervals from 0 to 5 hours in order to assess the small intestinal permeability, and from 5 to 24 hours to assess the large intestinal permeability. Concerning the results, here are shown the results from subject characteristics, and as you can see from the table, the two groups were similar for all general characteristics assessed, like gender, age, BMI, but also for the macronutrient composition of the habitual diet and physical activity level. As expected, because it was an inclusion criterion, the non-habitual milk consumer had a lower intake of milk compared to the control group. An interesting result then came from the analysis of the depression, anxiety, and stress scale questionnaire, and in particular, there was a higher frequency of anxious subjects in non-habitual milk consumer compared to the control group. Conversely, the depression and stress level were quite similar among the two groups. In this plot is shown the daily intake within each food categories, and as you can see, they were partly homogeneous, with the exception of dairy products, milk, and aged cheese that were higher in the habitual milk consumer, so in the control group, compared to the non-habitual milk consumer, which conversely had a higher intake of lactose-free milk. Concerning the intestinal permeability, the two groups showed a similar expression in urine of lactulose over mannitol and sucralose, suggesting that the small intestine permeability and the large intestinal permeability are not implicated in the milk-related gastrointestinal disease. So to conclude, we enrolled 39 healthy subjects, of which 20 were habitual milk consumer and 19 non-habitual milk consumer, and they were similar for all characteristics assessed, except for anxiety level that were higher in the non-habitual milk consumer. We found also some differences in the diet, in the habitual diet. In particular, the control group had a higher intake of milk, dairy products, and aged cheese compared to the non-habitual milk consumer. And finally, we found that gut permeability is not implicated in the milk-related gastrointestinal disease. The study is ongoing to evaluate in these two groups the relationship between the evolution of gastrointestinal discomforts upon milk consumption and the gastrointestinal hormonal response and the plasma profile of peptides coming from the milk digestion. Thanks for your attention, and I would like to thank also my lab group, in particular the lab head, my supervisor, the Professor Paola Vitaglione. Thank you very much.
Thank you, Silvia. Okay, you are live now. Congratulations for this innovative study and for the presentation. You showed that the correlation between diet and well-being in general is a quite complex topic. So now I can read some question that comes from the audience. From Fred Bruns, there is a question. The symptoms and related gastrointestinal symptoms can also be caused by wheat-related factors, gluten, amylase, trypsin inhibitors, fructans. Did you control for that?
So our volunteers just refer the gastrointestinal symptoms in relation to milk consumption. And we asked to them if they avoid some other food, but none of them refer these gastrointestinal symptoms with other foods. So thanks for the question.
Okay, but you check their diet. The only restriction was about the milk, or?
Yeah, we checked their habitual diet, and the only restriction was for due milk consumption.
Okay, thank you. I cannot see other questions, so I profit for a personal question from myself. Maybe there is a question from Uri. Do you think this can be linked to microbiota fermentation products?
Yes, could be. Indeed, we asked also to the volunteers to collect fecal samples. And indeed, the study is ongoing to check also the relation and the correlation with the gut microbiota and their functionality.
Okay, so we will know.
Yeah, we will know. Yeah.
Okay, my question was from what you observe. No, you observed that habitual consumers and non-habitual consumers had a higher anxiety score, if I understand. This psychological feature could be partly responsible for the discomfort.
Yeah, that's a good question. Thanks for that. Indeed, the bowel is known as a second brain. So there's a bidirectional communication between the intestine and the central nervous system. So the anxiety could be a predictor of the gastrointestinal discomforts, but meanwhile, it could be a consequence of that. So yeah, that's an interesting point to discuss with. However, as I said, it could be a consequence, but could be also one of the, could be also a causal role of that.
Yeah, I agree. There are two questions from two different persons, but they are related. One is where, I read both, were there some intolerant to lactose among the volunteers, and does lactose indeed cross the epithelial barrier intact?
Okay, so regarding the intolerance, yes, we found some intolerance volunteers. However, we exclude from our analysis, as I said before. And the second question was regarding the lactose.
If you observe that the lactose, if you check lactose cross the epithelial barrier intact or not.
Yeah, indeed, we are still analyzing data on urine samples because we also performed a milk test during which the volunteers drank 250 milliliter of cow milk, and then we collected urine at different time points, and we are still analyzing the lactose expression in this urine.
Okay. And there is another question that is again on lactose intolerance. Are the volunteers lactose intolerant or else are sensitivity to opioids, peptides? Okay, you already answered about the lactose intolerance.
Yeah,
and about the opioids?
Yeah, regarding the opioid peptides, that's a hot topic, and indeed there are several evidences suggesting the role of one of the opioid peptides, the beta-casomorphin 7, and indeed we are still analyzing again a dipeptide profile on plasma after a milk test in these volunteers.
Okay, and there is a comment from Maria Sotenko. What is shocking among 101 participants, 60% appear to be lactose intolerant. That shows the scale of the problem. Any comment?
No, we screened 101 volunteers, but not all of them were lactose intolerant. Some others we exclude for other reasons, because they have other pathologies, and we could not include in our study. Of some of them were obese, so that's why we screened a lot of so this larger sample size.
So lactose intolerance was not the only exclusion criterion.
No. This was not the only one exclusion criterion.
Okay, it was good to clear this.
Yeah, thank you for that question.
Thank you, Silvia. I think if there are no other questions, we can close this second session. I would like to thank all the speakers and all the attendants. Uri, want you add something?
Just wanted to add that perhaps it's also a good time for a nice cup of coffee. I don't know if I want it with milk or without after the seminar, but we have to take everything, with a half a happy face. Really, I take the opportunity also to congratulate Linda and Andre for an amazing event that, knock on wood, has been quite impressive in its outreach. Great work, you guys. I don't think that at Cork we will have the same numbers, so don't be disappointed. But I'm sure we will definitely have better food.
Indeed. So thank you, Yuri Lesmes and Pascal Ferrante, who came to us live from Israel and Italy, respectively, and indeed all the speakers who came from France and Portugal and Serbia, and what else did I forget? Italy, of course, that they all joined us live. And as you said, Yuri, we do want to thank the people who are joining us, especially on YouTube. Hi to Wabi Bajo, I think, from Mozambique, who's joining us on YouTube. And so this is the end of this session. So the next session will be on digestive lipases and lipid digestion, and it will start at 4:15 Irish time. So I think that gives us another little bit of time, as you said, Yuri, for coffee. So goodbye, and André is going to put up a holding slide until then.
See you guys.
Excellent. Just one remark down here, the menti.com. If you go to menti.com, fill in this code here for the location, and I have a second code for topics of the next Infogest webinar. So this slide will go on a loop, so it will stay there for a while, and we'll come back if you miss it. Okay. Good afternoon, Mern. Would you mind sharing the Mentimeter? I stop sharing my slides. Okay, so we actually see, here you go, that's interesting. So we have the numbers, but that gives you an idea from where people join. You see France, Ireland, you see Ghana, Finland, New Zealand, a few cities are here, Poland, India. So I leave that here for a minute. Colombia, and even Kilworth, which is right beside Moore Park. South Korea, Malaysia, that's great. So keep going to the Menti, there you go, to the Mentimeter. I just put it on again. Give me a second. That's down here. So the first number is for your location. The second one, I'm sorry. The second one is for the topic of the Infogest webinar. So we have another 5 minutes to go. I might as well introduce you, or you can introduce yourself, Frederic and Annabelle and Miriam, if you can put on your camera. So next session is session 3, and I hand over to you. You have seen the slides, so I suppose I won't go through the slides again, yeah? Is that correct, Miriam?
Yes, it is. Thank you, André.
Thank you, André.
Thank you.
Hello, everybody. Nice to see you again. So, Miriam, would you like to say a few words to start?
Yes, I wasn't really prepared, but yes. So as you saw on the slide, so the working group 4 is looking in lipids, lipolysis and lipase. So this is the topic of this session, and we've got a range of really good speakers with us today. So we will start with David Gray from the UK and the University of Nottingham. I can see you. Hi, David.
Hello,
and,
very nice to see you.
Nice to see you too. And we will have, so if I'm correct, we're having 2 PhD students, Susanna Pino and Olivia as well. Olivia was recently, completed our PhD actually. And we've got a bit of change in program. So we're having Sebastian Mars from INRAE, who will be presenting the last talk of today. So maybe Frederic, you can start and introduce our main speaker.
Thank you, Miriam. So my name is Frédéric Carrière. I am a CNRS researcher. I work at Marseille in France. And I have the great privilege to co-chair this session as well as the working group 4 of Infogest with Miriam and Annabelle that you can see on the screen next to me. So thank you very much for helping me to organize this session and selecting the presentation. So the first speaker today will be David Gray. So that's a great pleasure for us to welcome David as the first speaker. As Miriam already said, David is an associate professor in food lipid chemistry at the School of Bioscience of the University of Nottingham in the UK. He is interested in novel approaches of incorporating lipids, especially essential fatty acids and fat-soluble vitamins, into the diet to improve health, but also to reduce the impact of processes on the environment. In that context, he is looking for alternative and sustainable sources of dietary lipids. This includes studies on oil bodies, also named oleosomes, from plant seeds, but also chloroplastic fractions that can be prepared from edible plants, as well as from green waste from agriculture. I personally discovered David's work some years ago when I had to examine a PhD thesis at his lab, and I was fascinated to see how a rich source of nutrients, like a plant chloroplast, could be still recovered from a green waste, normally remaining in the field after harvesting. This research requires a better understanding of plant lipid digestion, and this is all about the presentation of David today. This fits perfectly with the topics we are covering within the working group 4 of Infogest. So I think this is now time to listen to David's presentation. So this presentation has been pre-recorded, but we will have time to discuss with David and answer to questions online after the presentation. Thank you very much.
I would like to start by thanking the organisers of the ICFD virtual conference for the opportunity to present this research. The work I'm about to describe needs to be placed in context, so please allow me to outline the work of my research group. In brief, my team and I are interested in establishing the properties of subcellular organelles as food or feed ingredients. These include oil bodies or oleosomes from oilseeds and chloroplasts from green biomass. Both are rich in lipids, and in both cases, our work includes optimizing their extraction, measuring their properties, and seeking applications. The properties we measure include composition, functional and physical properties, and the nature of their digestion. Not surprisingly, given the title of this talk, this presentation will only be concerned with the digestion of chloroplasts. Let me give a brief introduction to chloroplasts. These organelles make up about 20 to 30% of the dry solids of green leaf tissue and are striking in their elegant stacks of thylakoid membranes that provide a large surface area for the light reaction during photosynthesis. The approximate composition of a typical chloroplast is 50 to 60% proteins, 25 to 35% lipids, 5 to 10% chlorophyll, which is approximately 15% of the total lipid material, and a small amount of RNA and DNA. Given the topic, it seems wrong not to remind you of the life-giving reaction called photosynthesis. The light reaction on the left splits water and releases oxygen. The main products for the plant are ATP and NADPH. These molecules feed the dark reaction or Calvin cycle, where inorganic carbon dioxide is fixed into organic sugars. The process and biochemistry of photosynthesis has been studied since the 1600s, and modern science keeps revealing more and more of the beautiful intricacy of this process. Given our preoccupation with the mechanism of photosynthesis, few researchers have viewed these organelles from a nutritional perspective. This slide summarises the major nutrients found in chloroplasts in general. In terms of macronutrients, there are proteins and lipids containing essential amino acids and omega-3 fatty acids respectively. In terms of micronutrients, chloroplasts contain carotenoids, ascorbic acid, phyloquinone, tocopherols, and minerals such as iron. As noted in the previous slide, most of the lipids in the thylakoid membrane system are galactolipids, MGDG and DGDG to be precise. There are some species-dependent differences in the ratio of these galactolipids, but in general, DGDG is about 70 to 80% less than MGDG. Both lipids are enriched in alpha-linolenic acid. The precise abundance is affected by whether the plant is a 16-3 or an 18-3 plant. But even in the case of 16-3 plants, alpha-linolenic acid will make up not less than 60% of the overall fatty acyl groups. Before we move on to look at some results, let me justify the liberation of chloroplasts from green biomass. We have developed a simple physical process to do this. The benefits are it's a simple physical process, no solvents are used to extract the nutrients, and drying of this liberated preparation provides a natural, nutrient-dense material. Other benefits are likely, such as improved bioaccessibility of nutrients on removal of the cell wall barrier, and separation of nutrients from anti-nutrients that might be present in the plant cell. The potential applications include a functional ingredient in food and feed and a natural food supplement component. This slide shows the structures of lipophilic molecules in chloroplasts that have recognized nutritional value. So what role do they have in the chloroplast and where are they located? This kind of information is valuable when considering the digestion of macronutrients and release of nutrients from any food matrix or biomaterial. The light reaction of photosynthesis relies on a complex array of pigment molecules and electron carriers. PS1 and PS2 stand for photosystem 1 and 2. These are light harvesting complexes that contain many chlorophyll and carotenoid molecules within a mesh of proteins. Beta-carotene and lutein have a photoprotection role and improve light energy transfer in PS1 and 2. Electrons are passed on through a redox chain from PS2 to PS1 where NADPH is formed. Phyloquinone has a role in the electron transport chain associated with PS1. Alpha-tocopherol has a membrane protection role, and alpha-linolenic acid is a major building block of thylakoid membranes, which we've already covered. So here is the research question we're addressing. Do humans digest galactolipids when they eat intact chloroplasts? Research has been published on the hydrolysis of pure galactolipids by enzymes such as PLRP2, which can be found in the pancreatic juices from several animals, including humans. But this is the first study using intact chloroplasts. Here is a paper that emerged from addressing this question. And here is the PhD student, Jutarat, now Dr. Watanakul, who did all the work. Here are the other partners in crime. You actually may recognize some of them. What products of galactolipid hydrolysis might we expect during digestion? Based on published work from Frederic Carrier's lab, we would anticipate a sequential hydrolysis of fatty acyl groups, firstly from the SN1 position, to form the lysogalactolipids MGMG and DGMG, and ultimately we might expect to find these aqueous-soluble chemical species MGG and DGG devoid of any fatty acyl groups, along with a parallel increase in total free fatty acids. Now let me briefly outline the preparation of our chloroplast material and the in vitro digestion protocols we employed. We create a chloroplast-rich fraction by a simple physical process, as shown. Pea vine hilum is the field residue left after harvesting peas. For this work, both spinach leaves and PVH were heat-treated, and the CRF pellets from each were freeze-dried. We used a two-step static in vitro digestion model devised by the Carrier lab. This model uses pH 5 to mimic the gastric pH that most food in a normal meal would be exposed to, and rabbit gastric extract was included. Two sources of pancreatic enzymes were tested. In what we are calling experiment 1, we used human pancreatic juice, HPJ. Samples were taken across the whole incubation period. Experiment 2 had the same gastric conditions. The only difference here is that porcine pancreatic extract, PPE, was used instead of HPJ during the intestinal phase. After lipid extraction, samples were run on HPTLC, stained, and the amount of each lipid of interest quantified via densitometry. Here are the results of both the spinach and PVH CRF materials when exposed to human pancreatic juice in the intestinal phase. T0 equals the start of incubation, T90 the end. Overall, the pattern of galactolipid hydrolysis is very similar for both CRF preparations. Let's look at plates A and C. The amount of MgDG is reduced, with the emergence of MgMg caused by the loss of one fatty acyl group. Do we see a similar fate with DGDG? As with MgDG, after digestion we see a notable loss in DGDG. But there is no significant emergence of DGMG. This suggests that both fatty acyl groups are removed from the SN1 and SN2 positions. The increase in free fatty acids during digestion supports these observations. The next slide shows the rate of change of galactolipids and free fatty acids during digestion as determined by HPTLC results, and also includes the data from the porcine pancreatic extract experiment. These plots show spinach CRF data along the top, A and B, and P-vine howlum CRF data below, C and D. Incubations including HPJ are on the left, A and C, and those including PPE are on the right, B and D. You will notice from the time zero values that the spinach CRF has a higher amount of galactolipids per unit dry weight than the P-vine howlum CRF, but the trends for all the incubations were similar. Galactolipids are hydrolyzed mostly during the intestinal phase. An increase in MgMg, open circles, reflects the loss over time of MgDG, closed circles. A decrease in DGDG, triangles, mirrors MgDG loss in the presence of HPJ, but appears to be slower compared with MgDG loss when PPE is used instead of HPJ. Overall, the rate of galactolipid hydrolysis is faster in the presence of human pancreatic juice than porcine pancreatic extract. This final result slide shows the rate of free fatty acid generation in the 4 different incubations. Panel A shows the release of free fatty acids in controls without CRF material. It is clear from these results that the PPE extract itself contains some hydrolyzable lipids, as free fatty acids are generated in the intestinal phase. This was taken into account when calculating the amount of free fatty acids generated from CRF samples. Panel B shows spinach CRF, filled symbols, has less free fatty acid than the PVH CRF, open symbols, at the start. But during digestion, fatty acids are released from spinach CRF at a higher rate than from the digestion mixture containing P-vine howlum CRF. In the case of both CRF preparations, HPJ generates free fatty acids more rapidly than PPE. So here are our conclusions from this work. Galactolipids present in chloroplast-rich fractions from both PVH and spinach leaves are digested in vitro, mostly during the intestinal phase of digestion by pancreatic enzymes. Human pancreatic juice digests chloroplast galactolipids more rapidly than porcine pancreatic extract. This is probably due to the presence of PLRP2, that's pancreatic lipase-related protein 2, and CEH/BSSL, that is carboxyl ester hydrolase / bile salt stimulated lipase, in HPJ. Within the duration of simulated digestion in this work, HPJ promotes hydrolysis of the majority of MgDG and DGDG. The former is converted to MgMg, but DGDG appears to release fatty acyl groups from the SN2 as well as the SN1 position. After digestion, alpha-linolenic acid is the main free fatty acid from both CRF preparations. This work would not have been possible without university funding to allow Dutilat to visit Frédéric Carrier's labs in Marseille. And a big thank you to Terry for letting me drive a pea vine harvester and allowing us to collect the debris it left behind. Here are some pertinent references in this field of study. For a thorough coverage of galactolipids in nature and their digestion, I recommend the review noted at the foot of this list. Thank you for listening, and I'm happy to field any questions that you may have.
Thank you very much, David, for this very clear presentation.
Thank you.
I cannot see any question at the moment from the panel. So maybe Miriam and Annabelle,
you could fire away, Frederick, if you want. I don't mind.
So I had it's not really a question, but I know results from your lab that are really supporting what you have shown today about the digestion of galactolipid. And these are the dietary studies you already started in animal species. And I'm thinking about what you have done in the fish with Mohamed Ghedi. So maybe if you could say a few words about that, that would be great. Thank you very much.
Yeah, no, thanks, Frederick. So I think that publication is mentioned in that reference list on the last slide. So it's some work that was carried out by Mohamed Ghedi, one of my PhD students, now postdoc. And we were very interested in looking at the ability of replacing some of the fish meal in the fish feed with this green powder that we're generating, this CRF-rich material. And we found that the fish thrived on this replacement, this supplement, if you like. And not only did they thrive in terms of growth, there's some evidence that they preferred that feed. That's a small amount of evidence. But the most interesting thing that's relevant to this talk is that we measured the uptake of lipophilic molecules into the fish. And it was very clear that markers such as alpha-linolenic acid and 16-3, which is the triunsaturated 16-carbon fatty acid that is a signature of chloroplastidiol fatty acids, was taken up into the fish, as was the carotenoids, lutein, which got into the flesh and into the eggs of the female of the species. We also observed a conversion of beta-carotene into retinol. So we certainly saw in zebrafish very strong evidence of clear bioavailability of these lipophilics. So that's really exciting, and yeah, it's really, it's exciting work, and I'm glad that you picked up on it, Fred, and got excited too, which meant that we could collaborate and do this work.
Very interesting work.
Yeah.
So now, David, we start to have some questions on the chat. So I will start with the first one, which is by Lorenzo Nissen, who asks, what about microbiota? So I imagine it's about the action of microbiota on the galactolipids.
Yeah, it's a very good question. I think, obviously, if some of these chloroplastidial membranes and materials survive the small intestine and get to the large intestine, it is likely that they would be in a sort of fermentation soup. And I'm not aware of much work that's been done in this area, but I think it's an area that we'd love to explore more fully. There's certainly strong evidence of galactolipase activity in microorganisms, and you would anticipate that the galactolipids that do survive the small intestine, although this work suggests that quite a lot of it is digested, but any that does survive into large intestine, I think they'll be hydrolyzed into free fatty acids, and you'd anticipate that they might be further metabolized by the microorganisms that are present. So I think it's really interesting. And, we've talked about this, Fred, and the whole idea of what about the proteins in the thylakoid membranes? What's the fate of the proteins? No, I think it's work that we'd love to do. So absolutely. Good question.
So we have also a question by Olivia Ménard. So she said, PLRP2 and BSSL seem to be efficient for galactolipid digestion, but not present in the porcine pancreatic extract or at all level. What do you suggest to use for in vitro digestion model?
That's a good question. I suspect you've got the best answer for this, Frederick.
Yes, I have an answer. So,
well, why don't you answer it, Fred? You're the expert.
Okay, thank you, David. So I think many of you know that we are exploring novel sources of pancreatine within the working group 4. And we are looking for pancreatic extract that are more active than the one that are commercialized today. And we hope that among these new sources, there will be some that will be richer in PLRP2 and BSSL. And of course, we will have to compare them with the porcine pancreatic extract. And this is one of the tasks we have within the working group 4. So we have an information still by Lorenzo Nissen, saying that he is presenting a work on L minor or on a colon model. I'm not sure I understand really. So maybe Lorenzo, could you please precise what you mean? And there is an anonymous attendee who say, could you please comment on the emulsification properties of chloroplast lipids?
Yeah, I think that's a very good question. And there has been work done at Lund University, for example, on the functionality of thylakoid membranes and this satiating properties. I think that's fascinating work. And as part of that work, they've taken thylakoid membranes and observed the ability to stabilize an oil and water emulsion. So some of the work that we're currently doing is kind of addressing that whole question. It seems on one hand, we've got this ability within this in vitro model that's mimicking human digestion to actually break down galactolipids, but also there's evidence that thylakoid membranes seem to slow down the digestion of oils and fats. So I think the Lund University work strongly suggests that might be something to do with the photosystems 1 and 2, and that interesting collection of proteins and lipids that might be involved in slowing down the access of bile and colipase. So that's really interesting. So I think in terms of what's happening in the gut, I think there's a lot more that we need to study, to be honest. I think it's just an interesting tension between breaking down the galactolipids and the galactolipids being preoccupied at the interfaces of oil droplets in the small intestine. So yeah, it's fascinating. I'm not sure, I hope that answers the question.
I think so. So yes, I have a precision by Lorenzo Nissen saying that Flavia Casciano is presenting L minor, which is in fact duckweed. So we saw duckweed in one of your slides, David.
Yeah.
So they have done a work on the digestion of duckweed, and they invite us to look at the work they have done.
Oh, that sounds really interesting. Yeah, duckweed, fascinating, absolutely.
So this is the last comment or question I can see from now on. And I think that we have to move also forward for the other presentation. Thank you very much again, David, for your clear presentation.
Okay, pleasure.
See you soon. Bye-bye.
See you soon. Bye.
Yeah, so hi everyone. I'm pleased to present our next speaker, Susana Pinho, from the University of Porto in Portugal. She's in the last year of her PhD, and she's going to talk about the effect of cow's diet on the fatty acid release and oxidative stability during the gastric digestion of milk. So now we will see her pre-recording presentation.
Hello, my name is Susana Pinho, and I'm a PhD student in animal science from University of Porto in Portugal. My presentation is about semi-dynamic gastric digestion of conventional versus pasture-based milk, fatty acids release, and oxidative stability. I hope you enjoy. Our goal was to follow gastric lipolysis and lipid oxidation of commercial milks from the same species with different fatty acids composition. Because diet affects the composition of milk in fatty acids and the structure of the triacylglycerols. Gastric lipase preferential cleaves fatty acids at the SN3 position of the triacylglycerol, and physiological responses are modulated by fatty acid composition. We selected the semi-dynamic in vitro model described by Mulher Cabero because this model mimics gradual acidification, which is important for the activity of the gastric lipase. How physiological responses are modulated by fatty acids composition. Fatty acids contain different number of carbons, and depending on if they are short, medium, or long chain, their route of absorption will not be the same. But before absorption, fatty acids must be released from the triacylglycerols. Their digestion starts in the stomach, and free fatty acids up to lauric acid are readily used sources of energy because they are absorbed directly via the portal vein and are subsequently processed by the liver. Moreover, the hormone secretion in the gastro-intestinal tract is sensitive to the chain length of the free fatty acids. Specifically, long-chain fatty acids are potent inducers of cholecystokinin secretion, which is a hormone recognized to suppress appetite. In addition to lipolysis, the potential occurrence of oxidative degradation of fatty acids in the stomach cannot be ignored due to a probably lower bioaccessibility of unsaturated fatty acids than that expected. As previously referred, human gastric lipase preferentially cleaves fatty acids at the SN3 position. Moreover, this lipase cleaves primarily the short and medium chain fatty acids linkages. Milk has a unique profile of short and medium chain fatty acids, as you can see in this table. None of the other common lipid sources are constituted by butyric, caproic, caprylic, and capric acids. Importantly, these fatty acids are located predominantly at the SN3 position of the triacylglycerol, and thus gastric lipase plays an essential role in the hydrolysis of milk fat. Cow milk is the most common source of milk, and therefore it was selected. The fatty acids composition of milk is influenced by the lipid composition of the diet, and milk triacyl structure is influenced by milk fatty acids composition. Palmitic acid is the major fatty acid found in milk, and cows on pasture, eating fresh grass, produce milk with lower content of this fatty acid compared to cows under conventional management. The amount of a fatty acid at the secondary and primary positions is related to the amount of palmitic acid in the triacylglycerol. For example, please look at the graph. The proportion of oleic acid, the blue line, at the secondary position is negatively correlated with the amount of palmitic acid in triacylglycerols, and the proportion of myristic acid, the yellow line, at secondary position is positively correlated with the amount of palmitic acid in triacylglycerols. Moreover, fresh pasture feeding increased concentration of milk polyunsaturated fatty acids. These fatty acids prefer primary position, and as lipids undergo the hydrolytic process, the proportion of free fatty acids increases in the medium, and these have been reported to be more prone to oxidative degradation than triacylglycerols showing the same unsaturation degree. Now, what steps did we perform to respond to our goal? So, a milk brand with no label regarding their management and a brand with the label pasture-based were subject to the gastric phase of the selected semi-dynamic model. Then, the emptied samples were subject to lipid extraction using the Giblin method. To determine the fatty acid release, the free fatty acids were separated by solid phase extraction and analyzed by gas chromatography with flame ionization detection. To follow the development of lipid oxidation, the primary products of oxidation, conjugated dienes and trienes, were measured. How did we calculate the conditions for simulating gastric dynamics? We used the protocol supplementary information provided by the authors of the method. A real intake of 250 milliliters was considered, and the volume of milk subject to in vitro digestion was 15 milliliters. After filling out the spreadsheet, we got to know the volume and rate of gastric secretion, the total digestion time, and the time of emptying. We performed 5 gastric samplings, and immediately after each sampling, we added a mixture of chloroform and methanol to stop the activity of the gastric lipase. Then, we extracted the lipids from the gastric samples, performing the Giblin and Didier method twice. The water phase from the first extraction was treated with hydrochloric acid to reach a pH below 1.5. The organic phases of the 2 extractions were pulled together, and this extract is a mixture of free fatty acids, monoacylglycerols, diacylglycerols, and triacylglycerols. The free fatty acids were separated by solid phase extraction using aminopropyl cartridges. To separate the neutral lipids, the column was eluted with 4 ml of a mixture of chloroform and propanol. The free fatty acid fraction was then eluted with 4 ml of diethyl ether containing 2% acetic acid. Fatty acids were converted into fatty acids methyl esters using the acid transesterification process and analyzed by gas chromatography with flame ionization detection. The mass of each fatty acid was estimated by the chromatographic area of the internal standards triamcanoin or tricosanoic acid. Individual free fatty acids in each gastric sample were expressed as the percentage of the corresponding acylglycerol mass present in the undigested milk. To follow the development of lipid oxidation, the absorbance of conjugated amines and triamines was measured and extinction values were calculated from the ratio between the absorbance at 233 nanometers for conjugated amines or 268 nanometers for conjugated triamines and the concentration of the lipid solution in milligrams per milliliter. Now the best part, the results. The milk samples that we subject to simulated human gastric digestion were representative of milk from cows and conventional and pasture-based feeding systems. The pasture-based milk presented a distinct fatty acids profile, namely a lower relative content of palmitic acid and a higher relative content of polyunsaturated fatty acids, as you can see in the table 2. Conventional milk has 33% of palmitic acid, whereas pasture-based milk had 29%. Regarding to the content of polyunsaturated fatty acids, pasture-based milk had 5%, whereas conventional milk had 4%. Both milks had similar macronutrient composition, table 3, which was great because it allowed us to have equal conditions in the process of in vitro digestion. The change in pH during gastric digestion of both milk samples is illustrated in figure 2. The pH decreased in both samples, reaching a value below 2 due to the constant addition of gastric fluid containing hydrochloric acid. The low pH of gastric medium is an unavoidable pro-oxidant condition. Therefore, as digestion progresses, higher oxidative degradation of polyunsaturated fatty acids occur, as observed in the figures 3a and 3b. Milk containing more omega-3 fatty acids, such as the pasture-based milk, is potentially more susceptible to oxidation. Probably for this reason, the pasture-based milk presented a higher percentage of formation of conjugated amines than the conventional milk. On the other hand, an identical percentage of formation of conjugate amines was observed for both milks in all gastric sampling points, except for the last gastric sample, where a slight increase was noticed for the pasture-based milk. This similar percentage of formation of conjugate amines could mean a gradual progression of the oxidative reaction, since part of the conjugate amines are formed by further oxidation of conjugated amines. Now, the results of fatty acids release. Looking at the table, According to Zompasosa and co-authors, it would be expected a higher release of caprylic and capric acids for the pasture-based milk compared to the conventional milk, because the correlation between the amount of palmitic acid in the triacylglycerol and the amount of caprylic and caproic acids at the primary positions is negative. Therefore, a milk with lower content in palmitic acid, like the pasture-based milk, has higher amount of caprylic and caproic acid in the SN1 and SN3 position. Regarding to the lauric acid, the amount of palmitic acid in the triacylglycerol does not affect its positional distribution within the triacylglycerol. Looking at the graphs, the result of the capric acid was not expected. It would be expected a higher level of free capric acid for the pasture-based milk compared to the conventional milk. However, perhaps because of the complexity of the sampling, which is a process quite heterogeneous to protein coagulation, we were not able to observe this difference using this semi-dynamic model. Related to myristic, pentadecanoic and palmitic acid, it would be expected differences in the release of myristic and palmitic acid between milks. As you can see in the table, once the conventional milk had higher amount of palmitic acid, it would be expected a higher release of myristic and palmitic acid for this milk. As previously referred, the sampling process was quite heterogeneous, but another reason for not observing differences could be the fact that these fatty acids prefer the positions SN2 and SN1 of triacylglycerol rather than the SN3 position. In relation to stearic and oleic acid, it would be expected differences in the release of oleic acid between milks. As you can see in the table, for a milk with lower amount of palmitic acid, like the pasture-based milk, it would be expected a higher release of oleic acid. This fatty acid prefers the SN1 position of the triacylglycerol. Probably because of this, no differences were observed between milks. Considering the release of linoleic, linolenic, and the cis-9-trans-11 conjugated linoleic acid, our results were the opposite of the expected. For the conventional milk, we supposed a higher release of linoleic acid, while for the pasture-based milk, we expected a higher release of linoleic acid and the cis-9-trans-11 conjugated linoleic acid, according to the study about milk positional distribution conducted by Vallejo and Martin. The results of polyunsaturated fatty acids release were different from what would be expected, because probably they were oxidized. As you can see in the figure 3a, both milks presented formation of conjugated amines during semi-dynamic gastric digestion. In conclusion, the semi-dynamic in vitro model of gastric digestion described by André Brodkorb and co-authors was successfully applied to follow the gastric lipolysis and lipid oxidative degradation of milks with different fatty acids composition. Pasture-based and conventional milks presented differences in fatty acid release. Pasture-based milk presented a higher gastric release of caprylic and linoleic acid, whereas the conventional milk presented a higher release of linolenic and cis-9-trans-11 conjugate linoleic acid. The results of polyunsaturated fatty acid release were different from what would be expected, probably because they were oxidized. The similar percentage of formation of conjugate trains for both milks in almost all gastric sampling points could be due to the relative stability of conjugated amines, meaning a similar progression of the oxidative reaction for both milks. Importantly, our results indicate that physiological responses induced by gastric lipolysis of a pasture-based milk could be different than those of a conventional milk. Thank you very much, and I welcome your questions.
Thank you very much, Susana. It was a really interesting presentation, and it's also nice to see people using the semi-dynamic model. So we have some questions from the audience, and Menier asked, what is the conjugated fatty acid content of the different tested milks?
Yes, the content of the CLA was different from between milks, and the pasture-raised milk had higher amounts as expected.
Okay. We have another question from Marcos Ricardo Infantes Garcia. Could lipid oxidation be influenced by the open digestion system that permits oxygen to be in contact with the lipids?
Yes, we try to be careful, and we close all the open spaces with paraffin, but we need to remove the paraffin to take the samples. So could be, but in our stomach, in vivo, we also have dissolved oxygen. I know that it's different, but yes, the answer is yes, because it's a parameter that we cannot measure. We didn't measure.
Okay, another question from Marcos. To what extent fatty acid release from milk phospholipids could interfere with the quantification of fatty acid coming from triglycerides?
Yes, the fatty acid composition of the membrane, the phospholipids composition, affects the extent of the activity of the lipases. We didn't measure it, we didn't quantify and describe the phospholipids, but the truth is that the composition of fat globule membrane in phospholipids could affect that.
Okay, we have another question from William Kloen. Is sensitivity of oxidation of unsaturated fatty acids linked to its regional position? So the position of 1, 3 versus the second position?
Yes, PUFAs, unsaturated fatty acids, they prefer the primary position, so SN1 and SN3 position, and so they are cut by the gastric lipase in stomach, and as they are free, they are more prone to oxidation than if they are linked in the triacylglycerol.
Okay, we have a couple of questions more, but for the sake of time, I think we better move on. So if you can reply, typing the response, it would be great. So now we move on to the next presentation. So Miriam.
Yes, so it's my great pleasure to introduce Olivia Pavois. So she's, like I said, recently graduated from her PhD, and she's going to talk about the behavior of bulk aggregation of bile salt and the impact on lipolysis.
Hi everybody. I recently joined the team of Anwesha Sarkar as a postdoctoral research fellow to work on Aqualab, a European Research Council proof of concept funded project. But today I'm gonna show you part of the results I obtained during my PhD, which I carried out under the co-supervision of Cécile Dresse, Isabelle Gaillot and Yuri Girelli at both King's College London in the UK and the Institut Le Langevin in France. So my PhD project aimed at addressing fundamental questions on the role of bile salts during fat digestion, and my presentation today is focusing on the understanding of bile salts bulk aggregation behavior during fat digestion. So bile salts are biosurfactants produced in the liver and released into the small intestine, which play two key roles in fat digestion and absorption. So they promote enzyme-catalyzed lipolysis by facilitating the adsorption of the lipase-colipase complex onto fat droplet surfaces, and they also help remove insoluble lipolysis products from the interface by solubilizing them into mixed micelles. The origin of these contrasting functionalities or interfacial behaviors has been recently attributed to the small architectural variations of the bile acid portion, that is, the position and number of hydroxyl group, and not to the conjugating group like toro or glyco. More specifically, recent investigations carried out at the air-water interface, either bare or stabilized by a phospholipid or a polysaccharide film, have shown that cholate-based bisalts may facilitate the adsorption of the lipase-colipase complex, in contrast to deoxycholate and quinodeoxycholate derivatives that have been demonstrated to be more efficient at removing lipolysis products from the interface. So the objective of the work presented here was to bring insights into bisalts' behavior in solution in order to clarify how bisalt structure governs their peculiar bulk aggregation properties and how, in turn, this may explain their different interfacial behavior and contrasting roles during lipolysis. So bisalts are unconventional surface active materials which contain weakly separated hydrophilic and hydrophobic phases. This unusual planar polarity enables them to self-assemble in solution into micelles whose mechanism of formation and structure are still under debate despite extensive investigations. Our aim was therefore to provide a detailed molecular level picture of bisalts' complex aggregates and of structures relevant to the process of lipid digestion, thanks to the unique combination of scattering techniques and atomistic simulations. So as part of my PhD project, two distinct bisalts, sodium taurocolate and sodium taurodeoxycolate, which differ in the structure of their bile acid part and which exhibit different adsorption-desorption dynamics, were selected, and the bulk aggregation behavior was characterized. So first of all, we investigated the self-assembly behavior of each bisalt by assessing their micellization process with pyrene fluorescence spectroscopy, by resolving the morphology of the aggregates they form with small angle X-ray and neutron scattering, and by defining the molecular organization in their aggregates with molecular dynamic simulation. Thereafter, we studied the solubilization by bisalts of triacylglycerol incorporating liposomes with a small angle neutron scattering, and these liposomes were used to mimic ingested lipids. So first of all, we compared the aggregation properties of the two selected bisalts. Their micellization in water was studied, and their critical micellar concentration was determined by using fluorescence spectroscopy using pyrene as a probe to detect the onset of micellization. To do so, the evolution of pyrene emission fluorescence was monitored as a function of bisalt concentration. For each bisalt, 3 regions can be observed. So at low bisalt concentrations, bisalts are in the form of free unimers and pyrene emission is low, as it is in a polar environment. As bisalt concentration increases, bisalt's molecules start aggregating and incorporating some of the pyrene into a less polar environment. And at high bisalt concentration, a plateau characteristic of all the pyrene being fully inside micelles is reached. However, this study shows 2 distinct self-assembly processes for the 2 bisalts. So taurocolate exhibit a smooth gradual rise in pyrene emission fluorescence and therefore micellizes over a broad range of concentrations, contrary to taurodeoxycolate that displays a sharp increase occurring at low bisalt concentration. So these contrasting profiles are associated to different values of the CMC, which correlates with the hydrophobicity of the 2 bisalts, the CMC of taurocolate, the less hydrophobic bisalts, being higher than the one of taurodeoxycolate. Interestingly, these contrasting bulk aggregation properties can be linked to their interfacial behavior, which we investigated previously by injecting successive amounts of bisalts below a phospholipid monolayer formed at the air-water interface, and by monitoring over time the evolution of the surface pressure with the Lamre trough. So this study was performed at the phospholipid water interface in order to mimic the fat droplet surface stabilized by phospholipid molecules found in the gut. So an increase in surface pressure means that bisalts adsorb at the interface, whereas a decrease indicates desorption. Results clearly show that taurocolate remains adsorbed at the interface within a wide range of concentration, in contrast to taurodeoxycolate that demonstrates a higher propensity for desorption. Interestingly, for both bisalts, a common feature is that the onset of micellization in the bulk matches the onset of desorption from the phospholipid water interface. Indeed, below their CMC, bisalts' free unimers are able to adsorb at the interface, whereas the desorption process is triggered by micelle formation. This explains why a taurodeoxycolate desorbs at low concentrations, unlike taurocolate, which adsorbs over a wider concentrations range. Then, both small-angle neutron and X-ray scattering were used to characterize the structure of the micelles formed by the two bisalts at concentrations ranging between 2 and 200 mmol per liter, both in the absence and presence of salts. Various models were assessed, such as a sphere, cylinder, and ellipsoid, and the model that provided the best fit was the ellipsoid model. In the absence of salts, whose data are not shown here, the two bisalts exhibit a peak characteristic of electrostatic interactions. In the presence of salts, which corresponds to the data shown here, this peak disappears due to the screening of charges, except at the highest concentrations of bisalts, 100 and 200 mmol per liter. The structure factor was therefore neglected in the analysis of the small-angle neutron and X-ray data obtained with bisalts concentrations ranging between 2 and 50 mmol per liter in the presence of salts, which are the data I'm going to focus on during this presentation. Interestingly, larger aggregates were found by fitting the SACS data, which we can see in green here, compared with SANS data, which we can see in blue here. And this suggests that the micelles have a hydrated region or shell, which is probably not detected by neutrons. However, SACS data could not be fitted to a core shell model either, suggesting that this corona region is not well defined. Additionally, the extent of micellar hydration was found to be higher in the SACS data, in agreement with SACS probing this region better. Compared to taurodeoxycolate, much smaller objects are obtained with taurocolate, both below and above the CMC, probably because taurocolate has one more hydroxyl group and thus hydrophobic interactions driving bisalt self-assembly are probably weaker. For both bisalts, the micellar size was found to increase with concentration, suggesting a gradual process of self-assembly. This increase in micellar size is particularly notable in the lower concentration range, up to 20 mmol per liter for taurocolate and up to 10 mmol per liter for taurodeoxycolate. This shows that micellization takes place over a wide range of concentrations, particularly for taurocolate. Above these concentrations, the aggregation number stabilizes to 4 for taurocolate and to 11 for taurodeoxycolate. Such small micellar sizes are consistent with the peculiar structure of bisalts, where efficient packing is hindered by the planar shape of the hydrophobic region. Finally, molecular dynamics simulations were performed by two of our collaborators at King's College London, Rob Ziolek and Chris Lawrence, in order to obtain further insight into the molecular arrangement of each bisalts in their micellar aggregates at different concentrations in the presence of salts. As can be seen on these molecular dynamics simulation snapshots, on which colors are used to highlight either the different bisalts molecules or the different atoms, results show that bisalts molecules self-assemble via their hydroxyl skeleton, their ionic chain and hydroxyl groups being in contact with the bulk water. These observations confirms that bisulfite self-assembly is primarily driven by hydrophobic interactions occurring between bisulfite's hydrophobic faces, the hydrophilic moieties contributing much less to bisulfite's bulk aggregation behavior. After assessing bisulfite self-assembly properties, we modeled ingested lipids using triacylglycerol incorporating liposomes, and we investigated their fate in the presence of bisulfites in order to evaluate bisulfite solubilizing capacity. In this study, we used DPPC liposomes containing or not tricapryline or triolein as triacylglycerol. The effect of increasing amounts of bisulfites on the structure of triacylglycerol incorporating liposome, which we can see in black here, was investigated by small angle neutron scattering. Results show the presence of distinct structures in the 3 concentration ranges chosen. So at low concentrations, in blue, liposomes undergo very limited structural changes. In the intermediate concentration range, in dark green for taurocolate and in light green for taurodeoxycolate, the intensity decreases, suggesting solubilization of some of the vesicles into bisulfite micelles, while an interaction peak develops, suggesting intermicellar interactions. At high concentrations, in red for taurocolate and in dark green for taurodeoxycolate, the signal originates mostly from smaller interacting objects such as micellar aggregates, while most of the liposomes have disintegrated. The same faces are obtained in the same bisul concentration ranges for the other liposome compositions, so here DPPC tricapryline and here DPPC triolein, despite the different location of the triacylglycerol within the liposomes. The structure generated upon the addition of low and intermediate quantities of bisulfites were not analyzed due to the presence of multiple structures over a range of length scales. This would have required several assumptions to be made. However, the disruption of the liposome morphology is likely to be due to the adsorption or incorporation of bisulfite molecules into the vesicles bilayers, inducing then a gradual vesicle to micelle transition. The scattering from the aggregates formed at high bisulfite concentrations was fitted with the same ellipsoid model used for bisulfite micelles, and results showed that the mixed aggregates are larger. Than bisulfite micelles, and differences are more significant with taurodeoxycolate as compared to taurocolate. These results demonstrate that the addition of bisulfites to liposomes has a substantial impact on liposome structure, leading to their gradual solubilization into bisulfite mixed micelles, transitioning through various morphologies and their mixtures. Additionally, compared to taurocolate, lower amounts of taurodeoxycolate are needed to induce this transition. Indeed, a mixed micelle predominant phase is obtained at 50 mmol per liter for taurodeoxycolate compared to 100 mmol per liter for taurocolate. This correlates with taurodeoxycolate higher hydrophobicity. Therefore, this study provides evidence that taurodeoxycolate exhibits a better solubilizing capacity than taurocolate, which supports our working hypothesis that deoxycolate-based bisulfites are more likely to be involved in the incorporation of lipolysis products into mixed aggregates compared to colate derivatives, which instead may have a higher efficacy at facilitating enzyme adsorption onto the surface of fat droplets. To conclude, the study of bisulfite's bulk aggregation properties shows that the two structurally distinct bisulfites self-assemble via their hydrophobic phase into ellipsoidal micelles with their ionic chain and hydroxyl group facing the aqueous solvent. Our results also demonstrate that taurodeoxycolate forms larger micelles than taurocolate and is more efficient at solubilizing triacylglycerol incorporating liposomes into mixed micelles. All these findings confirm the significance so far largely neglected in the literature of bisulfite morphological richness in their bulk aggregation behavior and also support our hypothesis that taurocolate may be more prone to promote enzymes adsorption onto fat droplet surfaces, while taurodeoxycolate may be more efficient at removing insoluble lipolysis products from the interface through their incorporation into mixed micelles. Finally, I just would like to acknowledge the ILL for the PhD studentship provided and the beam time allocated on D33, as well as the ESRF and ISIS for the beam times allocated on ID2 and Sense2D. I also want to thank all my collaborators and supervisors for their contribution to the project, and finally you for your attention.
Thank you very much, Olivia, for this really nice talk and really nice work. So we've got a few questions. So Charlotte Oliver asked, what method did you use to evaluate the impact of bile salt on liposome?
So, as I said during my presentation, we used small longer nutrient scattering, and basically what we do is that we look at the structure of the liposomes, and then we see how bile salts will impact this structure. And what we found is that the more we had bile salts, the more we will obtain, the more we had bile salts, then we will see a transition from vesicle to mixed micelles. So basically these bile salts are just like disrupting the liposome structure and converting it into micellar aggregates, which we find were ellipsoidal.
Okay, another question from Mohamed Jedi. He asked, so first of all, he says thank you for the very nice presentation.
Thank you.
So do you have any recommendation of which one to use as a bile extract? Either porcine, bovine, or synthetic bile salt for in vitro digestion model?
Yeah, so from what I know from the literature is that so people are either using bile extracts, because I guess this is mimicking the best way what is really happening in the body. I also know that some people are using like a mixture of I think it was taurocolate and glycodeoxycolate, so one colate-based bile salts and one deoxycolate-based bile salts in a proper mixture so that they mimic what's happening again in the human body. So I would tend to say this is good because again, you have the two bile salts which we think have two different roles during lipolysis. So I would suggest to use either bile extract, which I have done for some of my in vitro lipolysis studies, or a mixture of both taurocolate and taurodeoxycolate, or taurocolate and glycodeoxycolate, for instance.
And this depends as well what you want to mimic, I think, because if you want to really mimic human in vitro digestion or match the human in vivo setting, we recommend at the moment, I think, to use bovine extract rather than porcine, because the proportion and the type of bile salt in those extracts are match a bit better the human one.
Yeah, okay.
I have a question, Miriam, if I can.
Yes, please.
So first, congratulations, Olivia, for your nice work on bile salts.
Thank you.
I have a question about, did you try to make similar experiments with a mixture of bile salts, mimicking the composition of the human bile?
No, during my during my PhD, I only focused on these two separately because we wanted to see their roles and their properties, or either interfacial or bulk properties. So no, I mainly used them separately. However, when I was doing some digestion studies as well, I was sometimes using bile extracts, but mainly because my objective was to look at both of them separately and understand their properties and roles during digestion. I just use them individually.
Okay, because when we see the difference between NATDC and NATC, you can imagine that the combination of the various of at least the 6 main bile salts will give two different results.
Yeah. Yeah.
Okay. Thank you.
And we've got another question from Sebastian Mars, who's going to present just after you, actually. What did you put tag in your system? It is not expected to solubilize in bile.
It's not expected to solubilize in bile. Yeah, so actually, because I think at first what we wanted to do, I mean, what we also did is adding both. So we added bile salts only and also bile salts plus lipase. So the other objective was also to study in the presence of both components. So in the presence of enzymes, yeah, we were expecting to see like digestion of this tag into MAG or fatty acid and then like mixed micellar formation. But yeah, for this presentation, I only showed like data with bile salts only. So yeah, you're right, it should not solubilize, but that's why we use them.
Okay. So I think we don't have any further question. And so I thank you very much again for your really nice presentation.
Thank you.
I think Annabelle, you're presenting the last speaker.
So following a similar topic, the last talk of the session is about the solubilization capacity of fat-soluble vitamins in bile salt micelles. This work was performed by Asma Elaud, a PhD student from INRAE in Nantes, France, but it will be presented by her supervisor, Sébastien Marcet, who will be also answering the questions after the talk.
Dear colleagues, I would like first to apologize because I'm obviously not Asma Elaud. I'm Sébastien Mars, his PhD supervisor. Asma asked me to make the talk, to record the talk for her because she was not able to do it. But she prepared all the slides, and this is obviously all our work. First, there is a difference. We know the difference between water-soluble vitamins and fat-soluble vitamins. There are a lot of different properties, of course, but also for absorption. It is more known for water-soluble vitamins and fat-soluble vitamins. This is because fat-soluble vitamin absorption is really linked to the digestion of lipids. And it's quite complex because they have to get incorporated in bile salts together with the digestion products of a triglyceride, or at least of lipids in general, and to form some what we call mixed micelles. And that's what we wanted to investigate because there's not a lot of data in the literature, or it's scattered, of the incorporation of these fat-soluble vitamins in mixed, what we will now call assemblies, because we are not even sure that it's really micelles. And to do that, we prepared some mixed assemblies with a constant bile composition made of taurocholate, lysophospholipid, fatty acid, and cholesterol. It's always the same composition. And in this bile micelle, because this is a bile micelle that we form with this composition, then we add different mixtures of fatty acid plus monoglyceride, which are the digestion products of triglycerides, the final digestion products. And we used either caprylic acid and monocapryline, or oleic acid and monoallene, or DHA and MDH at different concentrations. You can see we started from 5 up to 40 milligram per milliliter in the sample. To characterize the assemblies that the forms, we use different experiments, DLS, dynamic light scattering, to measure the size distribution of spherical objects in the range 1 to 1000 nanometer. We use also a spectrophotometry at a wavelength of 600 nanometer. to make a turbidity measurement, to investigate the macroscopic transitions in the sample. And we use also SAX, small angle X-ray scattering, which gives not only the size in the range 1 to 100 nanometer, but also the shape and the structure of the assembly. The principle is that you make a scatter of the X-ray and you get an intensity profile as a function of the wave vector, and this wave vector in fact is the inverse property of a length scale, so you can derive some length scale in this profile. And using modeling, you can resolve the real assembly structure that you have in your sample. It can be micelles, spherical, vesicles, long cylinders, can be lamellar, planar structure. So you have different kind of structure, especially in these systems where you have a lot of surfactant molecules. We first will show the results for mixed assemblies. The mixed assemblies are these bile compounds with increasing concentration of fatty acid plus monoglyceride. And you can see in the blue curves first that for caprylic acid there's nothing really going on. It's the diameter of the assembly is always constant and is about 3 to 4 nanometer, which is typical for a micelle size. However, for oleic acid or DHA, you can see that there's a size transition. It goes from a typical micelle size, 4 or 5 nanometer, up to, in the end, a size around 22 nanometer, which is more typical of a vesicle size. For the turbidity, you can see that we have the same behavior. For the caprylic acid, there's nothing going on. The samples are transparent and there's no transition. But for oleic acid and DHA, the sample turbidity increases as the fatty acid concentration increases, which show that there is also a macroscopic transition that you can see by eye also. Now for the SAX results, you have this intensity profile as a function of this wave vector, and you can see in red these are the experiments, and in black the black lines are the models. So you can see that there is a difference in the shape of this intensity profile as a function of the concentration. When you go up in the curves, in the graph, the concentration of the fatty acid is increased, and you can model. In fact, we did the model of these SAX profiles using only one model. We use a lot of models, but the best one was a model that is based on a water core and multiple shells around this core, and a sphere model. So we used the SESView to fit these models, and we found that for oleic acid and DHA at high concentrations, we found a water core and 3 shells around this core. And for caprylic acid and the low concentration of the other fatty acids, we found that there was no water core, in fact, and that there was 2 lipid shells. So we plot the parameters of this model as a function of the concentration of the fatty acid, and we found that there was nothing going on for the caprylic acid, which makes sense. It's the same for DLS, and we also found that the micelle was composed of 2 regions: the yellow one, which is the region for where the lipid tails are, and the green one where it's the region for the lipid groups and the surface molecules, for example for bile salts, which are at the surface of the assemblies, and we know that they are in a transverse position. not a typical surfactant as we know it. Then we were looking at the oleic acid and DHA systems, and here we can see a steep transition in these parameters. And we saw that with the model that you had a transition between micelles and vesicles. And we can see three shells around the vesicles. These are not multilamellar vesicles. These are unilamellar vesicles, but the shells are the same as for the micelles. It means that there are some region for the tails, the lipid tails, and region for the head grafts. We checked these results with a cryo-TEM, so electronic microscopy, and we saw that for a small concentration of oleic acid, we can see only points, small points in the observation. Those are the micelles, and when you go to high concentrations of oleic acid, then you can see that there are some vesicles appearing. And they are not multilamellar. They are indeed unilamellar, but some of them have vesicles entrapped in vesicles. For DHA, it's the same. We can see the same behavior. A little bit smaller vesicles are obtained. Now we want to investigate the incorporation of fat-soluble vitamins in these mixed micelles or vesicles that we found. So we selected two concentrations, the lowest one in fact and the highest one, to investigate this incorporation. We used all the fat-soluble vitamins, a specific molecular form of, and we test a very large ranges of vitamin concentrations addition into the assemblies. The results are that we wanted to measure a solubilization capacity in these mixed assemblies, and first we did it for alpha-tocopherol, vitamin E, or phylloquinone, vitamin K, in caprylic acid mixed assemblies. So you know that for caprylic acid it's only micelles. And you see the size of the micelle at some point. It will have a steep increase measured by DLS, and this steep increase is defined as the solubilization capacity. And we can see in these results that is higher in mixed micelles containing more caprylic acid. The blue points are always the increase is always at higher concentration of vitamin. It's the same for vitamin K. Now we wanted also to see what's going on in vesicles. So we used also increasing concentration of here in this case it's vitamin E in oleic acid mixed assemblies. And we did the case for the low concentration of oleic acid, which is the micelle, and the high concentration of vitamin, which is the vesicle. And you can see that the transition point is not at the same place, depending on the assembly that we are investigating. And we found that the solubilization capacity is higher in vesicles compared to micelles. Now we did that for a lot of, as you saw, we did that for all the systems that we investigated. So it means that we did for all the type of fatty acids plus monoglyceride, but also we did it for the two concentrations, so which are 5 and 40. And so for except for caprylic acid, of course, we had some systems with micelles and others for vesicles. And in fact, what we found for vitamin E and vitamin K is that the solubilization capacity depends largely on the fatty acid plus monoglyceride concentration, but less on the fatty acid plus monoglyceride type. Now we did the same also for the cholecalciferol, so vitamin D. And here we are looking at the oleic acid system. So we have either micelle or vesicles. And here at the left graph, you can see the diameter as a function, the diameter of the assembly as a function of the vitamin D, and you can see that in a very, very large range there is nothing going on, and we have to go very, very high to start to have an increase, but it's not even a very large increase or steep increase compared to the increase that we had before for the other system. And for the vesicles, so for the high oleic acid concentration, we don't see any increase at all, even when we go to very high concentration. And this confirms that the vitamin D is a very special vitamin that can self-assemble, that it was found previously a few years ago. In conclusion, adding oleic acid or DHA at high concentration changes the structure of mixed bile micelles. Micelles are indeed formed only at low concentrations, whereas vesicles are formed at high concentrations. Adding caprylic acid does not change the structure of mixed bile micelles. Sachs results show that micelles are spherical or slightly ellipsoidal, and they have two regions, and they show also that the vesicles structure is made of three shell regions. The solubilization capacity of vitamin E and vitamin K increases with fatty acid concentration, but whatever the type of fatty acid is. And vitamin D induces no structural transitions in the assemblies, likely due to its self-assembly. Thank you for your attention.
Thank you very much, Sebastian, for the nice presentation, and thank you, Asma, for the work. We don't have any questions from the audience yet. I have a question myself, so I might have missed it, but there was any rationale to select those specific fatty acids and monoglycerides?
Because they were very different in terms of structures and in terms of solubilization properties. In the literature, we saw that in bile salts there are some different solubilization capacity already in simple bile micelles of these fatty acids, and also because we selected this specific fatty acid and monoglyceride because they all are very important for nutritional purposes. That's why we wanted to investigate those ones, because they are very specific and have very different properties.
Okay, that's good. I have another question. So what is the mechanism for that enhancement of the fat solubilization of vitamins? Sorry, what is the enhancement of that fat soluble vitamins when there is an increase of fatty acid and monoglycerides in combination?
Yeah. It's always a combination of fatty acid and monoglyceride, but what we observe is these structural transitions, and we are not sure 100% for now that it's a structural transition that drives the incorporation of vitamins. But what is sure is that when we increase the concentration of fatty acid and monoglyceride, there is an increase of this incorporation of vitamins. We have to make sure now that it's not due to the amount of lipids incorporated, but really due to a structural effect.
Okay. Well, we don't have more time. Thank you very much, Sebastian. So with this, we conclude the third session of the conference. We would like to thank all the speakers of the session for the great talks. Before finishing, I think André would like to say a few words. So over to you, André.
Not too much, but I would like to thank, first of all, the session chairs and all the speakers of today. We were up to 365 attendants, and we had a couple of them on top of that on YouTube. Please remember that all talks were recorded, and they will be uploaded onto the YouTube channel afterwards, so you can watch some of them again, or if you missed any talk, you can watch it. So I don't have much to say really. Usually at this point, we move slowly to the bar, might have a drink or something small to eat. So treat it like a real conference day. Do just that, you know, might have something small to eat or something small to drink, or whatever suits. And we see you again in 6 o'clock, which is about in 20 minutes time. There's a 2-hour poster session organized by our postdoctorates. And if you can wait to the very end, you can actually vote for the best poster. Okay, very good. Linda, do you have anything to say there?
No, I'm all good. So thank you everybody for tuning in for the whole time. I know it was a long day, but hopefully we'll see all the poster sessions next.
Excellent. And otherwise, see you tomorrow morning, 7:30 Dublin. So early start for us, might be a late start for others. It's a live session from Australia and New Zealand. Okay, all the best. Bye.
Thank you very much, Linda, André, Annabelle, Miriam. Bye-bye.